A very good morning to the galaxy of intellectual present here. With the blessing and encouragement of Honorable Founder President Dr. Ashok K. Chauhan sir, Honorable Chancellor Dr. Atul Chauhan sir, Honorable Senior Vice President, Amity Education Group, U. Ramachandran sir, Honorable Vice Chancellor Professor Dr. Raman Kumar Jha sir, Honorable Vice President Gaurav Gupta sir, respected Director AUJ Professor Dr. Ajit Kumar Pandey sir, respected Registrar Prabhakar Tripathi sir, and respected Assistant Registrar Prabhat Kumar Tripathi sir. We are organizing two-day international conference on the topic Challenges and Opportunities in Biotechnologies. Welcome you all. As a mark of respect for our mother nation, we start any event with the national anthem. So now I request Dr. Amit Kumar Datta sir to kindly play the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
We at Amity University are known as custodian of culture, and continuing the tradition, we shall now seek blessing by lighting the lamp virtually. I request our technical team to prepare for the lamp lighting.
भर्गोदेवस्य धीमहि धियो यो नः प्रचोदयात्। शारदे मां अज्ञानता से हमें तार दे मां। मुनियों ने समझी है गुणियों ने जानी, वेदों की भाषा पुराणों की वाणी। मुनियों ने समझी है गुणियों ने जानी, वेदों की भाषा पुराणों की वाणी। हम भी तो समझे, हम भी तो जाने, विद्या का हमको अधिकार दे मां। हे शारदे मां, हे शारदे मां, अज्ञानता से हमें तार दे मां। हे शारदे मां, हे शारदे मां, अज्ञानता से हमें तार दे मां। हे शारदे मां, हे शारदे मां, अज्ञानता से हमें तार दे मां।
Moving forward, I would now request the convener of the two-day international conference, Dr. Amit Kumar Datta, sir, for his introductory remark. Sir, please.
With the blessing of our Honorable Founder President, Dr. Ashok Kumar Chauhan, sir, our Honorable Chancellor of Amity Education Group, Dr. Atul K. Chauhan, sir, I, Dr. Amit Kumar Datta, from Amity Institute of Biotechnology, Amity University, Jharkhand, India, welcome you all on this virtual platform for the inaugural program of 2 days international conference on challenges and opportunities in biotechnology, ICCOB 2021. Blessings from our Honorable Vice Chancellor, Professor Dr. Raman Kumar Jha, sir, respected Director, Professor Dr. Rajit Kumar Pandey, sir, today's invited Chief Guest, Dr. Mrs. Kalaivani, Head, Department of Basic Science, Faculty of Health Sciences, the Open University of Sri Lanka, invited distinguished guest, Dr. Aarti Shanware ma'am, Director, Rajiv Gandhi Center for Biotechnology, RTM Nagpur University, Nagpur, invited guest and very own our respected Registrar of AUJ, Mr. Prabhakar Tripathi, sir, Organizing Chair and University Coordinator R&D Cell, Dr. Jaita Chattopadhyay ma'am, and all delegates and dignitaries from India and abroad, and all esteemed colleagues and organizing committee members. A very good morning to one and all from India. Biotechnology is one of the promising fields of biology that involves application of scientific and engineering principles to the processing of material by involving biological agents. The 2 days international conference on challenges and opportunities in biotechnology at Amity University, Jharkhand, Ranchi will be involving multidisciplinary fields of biotechnology, providing opportunities for exchange of ideas, scientific knowledge and experience among researchers, academicians, scientists, and students from different fields of biotechnology. The scientific program will include plenary as well as keynote lectures along with oral and poster presentation under different themes based on sustainable principles, ideologies, and practices involved in biological sciences, computational approaches in biotechnology, microbial and agricultural biotechnology, as well as upcoming technological advancements. The main aim of this international conference is to bring together leading academicians, eminent scientists, researchers, and scholars from different universities and institutions of world to exchange and share their experiences in recent innovations as well as challenges in biotechnology. This 2 days international conference will provide an international platform for discussion of recent advancements, present, and future challenges in biotechnology. On this beautiful sun-shining day, by the blessings of our respected Senior Vice President, Mr. U. Ramachandran sir, Honorable Vice Chancellor, Professor Dr. Raman Kumar Jha sir, and respected Vice President, Mr. Gaurav Gupta sir, as a convener of this today's international conference, I hereby declare the international conference is open. Over to organizing session chair member, Dr. Nimi Srivastava ma'am, for further process.
Thank you, sir. Now, I would take the privilege to welcome our respected registrar, sir, to come forward and mark the beginning of today's international conference with his elite speech. Sir, please.
I request to please unmute me. Am I audible?
Yes, sir. Audible, sir.
Good morning. With the blessings of Honorable Founder President, sir, Honorable Chancellor, sir, Honorable Senior Vice President, sir, Honorable Vice Chancellor, sir, respected Vice President and Director, sir. I once again welcome all the dignitaries and the renowned speakers from all over the world-renowned institutions like Sri Lanka, Poland, USA. This is a great opportunity that our Department of Biotechnology and conveners are thinking that what are the opportunities in biotechnology. So organizing such wonderful conference that gives a good vision of biotechnology department, and the last 1.5 years has given understanding that the Department of Biotechnology, I mean to say biotechnology, has more opportunity in comparison to now challenges. Because in all this pandemic, it is only the one field which is biotechnology. All the scientists, even students, faculty members, and scientists, they have contributed a lot in biotechnology. And this is the scope of biotechnology, that these students can understand what the future demands from them, demands from scholarly brain of biotechnology students, faculty, and all scientists, field experts. And I'm sure that this wonderful international conference is going to contribute to all the students, faculty, and also other interest, having interest in this field. So kindly accept my heartiest welcome and greetings on this international conference, and my best wishes to all participants in this wonderful organization. Thank you, Dr. Amit Datta. Thank you, Dr. Nimi, and all the whole team of this international conference organizing team. Thank you.
Thank you, sir. It's always very nice to listen to you. Now, moving forward, I would like to first extend my warm welcome and regards to our chief guest who have joined us for the two-day event, Dr. Mrs. Kalaivani ma'am and Dr. Aarti Shanwari ma'am. Welcome, ma'am. And now I would like to request Dr. Kalaivani ma'am first to please acknowledge the audience with her fountain of knowledge. Ma'am, please.
Good morning.
Ma'am.
Want to switch on the video?
Yes, ma'am.
The talk will be starting 11 o'clock, no? 11:40?
Yes, ma'am.
Yeah.
Kindly give your blessing for us to start the event, ma'am.
Okay. Okay. Good morning, everybody. It's a great pleasure to be all with you all at this moment at the International Conference on Challenges and Opportunities in Biotechnology. It's a great pleasure to me to also to coming as a chief guest to deliver the this keynote address. So actually, this is a great opportunity and great thing that you all have invited from Sri Lanka. So that's the neighboring country. That is a with the I pray for your the best thing that the international conference has to be in a great success, and also wish all the delegates and all the participants to start this event with a great success. Thank you.
Thank you so much, ma'am. The second notable women scientist with us today as the chief guest is Dr. Aarti Shanwari, ma'am. I will now request ma'am to please give us blessing to start the event.
Thank you very much, Dr. Nimi, ma'am, and a very good morning to you all. It's a moment of great privilege and honor for me to be invited as a panelist today and later tomorrow as a guest speaker. Coming today at this inaugural event for which we have gathered for 2 days international conference on challenges and opportunities in biotechnology, ICCOB 2021. On 11th and 12th November 2021, organized by Amity University, Amity Institute of Biotechnology to be precise, Ranchi, Jharkhand, through virtual mode. Further, this event holds special value due to the changing time caused by the COVID-19, where the entire proceedings will be taking place on a digital platform. I please accept my hearty greetings on this notable occasion once again to our internationally acclaimed distinguished guest for today's event, other dignitaries, my learned colleagues from various university departments across the country, delegates attending the webinar, and students' friends. As you all are aware that we are very fortunate to be a part of this 2 days academic feast to celebrate and popularize science and its application among students, to foster the spirit of scientific temperament and to stimulate their minds to explore new frontiers in biotechnology, to display all the activities, efforts, and achievements in the field of science and for the human welfare as well, to discuss all the issues and implement new technologies for the development of the science, and to give an opportunity to the scientific-minded citizens in our country. It is very well said that the aim of education is the knowledge not of fact but of values. It is on such occasions, I feel, that we get opportunities to test our knowledge and understanding. We look forward to getting exposure to what the best of their brains think about this very dynamic and equally wonderful field of life sciences. We, as a mentor, as a teacher, as a guide, are committed to broadening awareness about life sciences, particularly in biotechnology, by learning how students we inspire and lead to the development of other innovations as well. Anticipating the effect of such issues is therefore as imperative as advancing its capabilities, and in align with this backdrop, I again feel very honored to be present in the company of great scientific icons at the international level. Just as houses are made of stones, so is science made of facts. But a pile of stones is not a house, and collection of facts is not necessarily a science. That is what I feel. Only someone with great and deep insight and a clear vision of the future, like all the erudite resource persons of today's and tomorrow events, can bring forth the hidden gems of wisdom to the surface. We are very grateful to all of you, sir and madams, for your august presence and your views as well. I don't think there is anyone who doesn't recognize the importance of research and development as a crucial ingredient in stimulating innovations improving the levels of productivity, creating added values, and thereby generating economic growth of the country. Therefore, this particular event is a potential platform to realize the need of upgrading innovative research, particularly specifically in biotechnology. Before turning to the safety and security of biotechnology research and the need to balance risk mitigation with innovations in the field, it is important to understand the scope and application of such technologies. Therefore, biotechnology, as defined by the Organization of Economic Cooperation and Development, refers to the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. Some applications of biotechnology include developing food products, biochemicals, beverages, and pharmaceuticals, waste management, and sustainable resource procurement. Scientific development in biotechnology can offer solutions to address global challenges such as combating the spread of infectious diseases, reducing hunger, and remediating environmental degradation as well. Biotechnology has the potential to create novel diagnostics, vaccines, drugs, and other medical countermeasures needed to detect, prevent, and treat infectious diseases. For example, recently we are facing the coronavirus pandemic has highlighted the promising role that biotechnology can play in this way. My learned speakers before my talk has already highlighted this very point. And in addition, biotechnology can be used to create genetically engineered organisms that can be deliberately introduced into the environment. As an example, advances in genome engineering technologies such as clustered regularly interspersed short palindromic repeats, that is CRISPR, and CRISPR associated protein 9, that is Cas9, have enabled the development of gene drives, a technology that allows desired genetic alterations to spread faster through a population over many generations. Agriculture biotechnology, which is very core to my heart and very near to my heart as well, can be used to create genetically modified crops for combating hunger and malnutrition. Traditional biotechnological techniques such as selective breeding, hybridization, and fermentation have been modifying living plants for improved yield or enhanced nutritional value from time immemorial. However, with improvements in knowledge about the role of individual plant genes, modern biotechnology techniques can be used to add, delete, or edit specific genes to produce a desired variety, thereby reducing the possibility of off-target effects. Scientific advances can also help develop genetically modified crops that withstand natural calamities, pests, and diseases. Such varieties can increase crop yields, lessen the need to use chemical pesticides, insecticides, lift farmers out of poverty, and ensure food security. That is the major point. Beyond that, biotechnology can help produce healthier and fast-growing animals and improve the quality and quantity of milk, eggs, and meat for human consumption. Environmental biotechnology has the potential to help mitigate pollution by using microbes and their byproducts. Instead of chemical methods to treat solid, liquid, and gaseous waste, plastic pollution, one of the most pressing environmental concerns, can also be addressed through biotechnology. For example, some bacterial enzymes can digest the raw material used to produce single-use bottles for beverages. Moreover, bioplastics can be produced with renewable raw materials such as plants, vegetables, and other recycled forms of waste that can be environmentally friendly alternatives to oil-based plastics. Real-world applications are integral to the successful development and use of biotechnology. In the pharmaceutical industry, for instance, streamlining the process of getting new treatments to market, the translational medicine standard procedure for improving that new treatment is safe and effective include animal models and clinical trials. For environmental biotechnology, the process of getting products to market have not been established yet. The application of environmental biotechnology often fall outside of existing regulatory framework. Biotechnology has the potential to revolutionize the societies that humans live in, the organisms that they live alongside. Recent advances include cheaper and more accessible DNA sequences, faster DNA synthesis, discovery of efficient and accurate gene editing tools, such as developments in synthetic biology. Meanwhile, there is a growing do to yourself, that is DIY community of independent biotechnology practitioners and enthusiasts who are unaffiliated without any single lab. Despite their promise, developments in the field do raise certain safety and security concerns as well that policy makers and scientists must bear in mind. It is therefore important to evaluate global best practices, treaties, and conventions that deal with such risks and devise strategies to update the safeguards to govern dual-use applications of emerging biotechnologies. Continuing advances in biotechnology provide a plethora of opportunities to address global challenges such as spread of infectious diseases, food security, environmental degradation. However, the same technologies can be deployed by some hostile states to create deadly pathogens that deliberately can cause human infections, negatively target agricultural supply chains, or disrupt existing ecological balances. To tackle such challenges, most countries have adopted informal guidelines or laws to ensure the safety of biotechnology-related research, instituted mechanisms to prevent unauthorized access to biological materials, and create export controls to govern the transfer of sensitive biological materials. Moreover, there are no mandatory global standards on biosafety and for the all research laboratory, I must say, must abide by, and there is no mechanism that introduce accountability and proper procedure for judging claims of liability when experiments go away. For example, when the above examples highlight that these global mechanisms are ill-equipped to handle threats emerging from breakthroughs in biotechnology. To ensure that this related research is conducted responsibly, the international community needs to collaborate to develop standards that govern the safety and security of experiments, formulate the long debated verification and monitoring mechanism under this treaty. Famous philosopher and essayist George Santayana has once said that technology is nothing but developed perception, interpreted intent, and common sense rounded off and minutely articulated. It is fact that science gathers knowledge faster than the society gathers wisdom. And when scientific knowledge is crowned with wisdom through decades of experience, the combination commands deep reverence. I once again thank you for patience listening and inviting me as a panelist and later as a eminent speaker or invited speaker. And I'm very thankful to all your organizing team and wishes my best for your successful conduction and other proceedings as well. I'm sure that the expert talk, which will be delivering in 2 days, this academic feast, will be delivered in the forthcoming session by eminent speaker, will bring you the feast of knowledge for stepping towards innovations, safety, and to strengthen the industry academia interface as well. Thank you all. Have a nice day ahead. Thank you so much, one and all.
Thank you so much, ma'am, for your motivating speech. Now, I would take the privilege to welcome and request one of our invited guests, Dr. Vinay Kumar sir, to kindly share few words in this inaugural session. Dr. Vinay Kumar sir. I would request Dr. Vinay Kumar sir to kindly share few words in this inaugural session.
Maybe there will be some network glitches, ma'am. So kindly please, ma'am, proceed for further.
Okay, sir. The Research and Development Cell in charge, Dr. Jaita Chattopadhyay, ma'am, is with us. So I would request ma'am to kindly share a brief introduction about the research and development activity undertaken at Amity University, Jharkhand. Dr. Jaita, ma'am.
Thank you, Dr. Nimi, ma'am. Good morning, our respected registrar sir, Prabhat Kumar Tripathi sir, and all the faculty and participants and speakers. And it is a great initiative from Amity Institute of Biotechnology. Of course, in today's world, we cannot think any domain of study without considering biotechnology. And at this moment, I think basically biotechnology and nanotechnology fields are booming all over the world. So of course, this conference will definitely spread great knowledge among all the participants and students, whether they are directly from this domain or not. I welcome all the respected speakers and participants to this esteemed conference. I extend my best wishes to the organizers and all the team members. Of course, we will be waiting for such great events at Amity University, Jharkhand in near future also. Thank you. All the best.
Thank you, ma'am. I would now again request Dr. Vinay Kumar sir to kindly share his words. If I'm audible, Dr. Vinay Kumar sir, am I audible? I guess that sir is facing some technical glitch. So moving forward, with the theme opportunities in biotechnology in mind, we would now like to present a short video in front of you, having all the glimpse of the opportunities undertaken in the field of biotechnology. So over to Amit sir, kindly please share the video.
Welcome to Amity Institute of Biotechnology. The discipline of biotechnology has made its presence in all spheres of life. It has found application in agricultural sector, medical field, with most promising area in vaccine development, bringing innovation in textile industry, food and beverage sector, and many more. With its immense scope, the team of teachers and students at Amity Institute of Biotechnology is focused in addressing the nation's challenges by contributing in the discovery of biology and applying the knowledge for the welfare of the people of Jharkhand and India as a whole. In this connection, we all are committed in nurturing young minds and producing highly trained graduates of BTech Biotechnology and BSc Biotechnology at Amity University, Jharkhand.
We at Biotech Department, having diversified and experienced doctorate and postdoctorate faculty members in the field of stem cell and tissue engineering, precision medicine, gene editing, synthetic biology, plant and animal biotechnology, enzyme technology. Our brilliant teaching staffs are really giving their innovative ideas and sharing their knowledge to create and develop researchers, scientists, entrepreneurship and academicians who will improve the quality of human life and contribute the society by producing responsible, competitive, budding candidates in the field of biotechnology.
With vision of creating Center of Excellence in frontier area of research, here at Amity University Jharkhand Biotechnology Department, we undertake application-oriented research and extension activity to make a long-lasting contribution to the advancement of scientific knowledge with active involvement of qualified faculties and students.
Biotechnology provides a whole new world for research and various experiments. There are so much research going on worldwide that improve the industrial yield. For example, mRNA vaccine production by biotechnology, which is a revolution to the vaccine industry, preparing nanoparticle for industrial scale. Biotechnology are also involved in textile industry, in pharma industry, in food industry, in agriculture, and in computing information. Amity Institute of Biotechnology, Jharkhand has association with many industrial partners to narrow the gap between industry and academia and also to nurture the student skill to meet up the requirement of the biotech industry.
Biotechnology is the branch of science that utilizes living organisms and techniques to develop products and processes that are useful for mankind. Presently, India is one of the top 12 biotechnology destinations worldwide. There are various government organizations in India like DBT, BIRAC, BCIL, ICMR and many more that supports research and development in the field of biotechnology. By 2025, India will be contributing in the world biotechnology market by 19%. So biotechnology is emerging as a very nice career aspect. We here at Amity Institute of Biotechnology nurture these students in such a manner so that they can fit in the various career aspects of biotechnology worldwide.
I am coordinating training and placement in biotechnology. You can join either BSc or BTech program here, especially for those students who could not crack NEET or JEE examination. They can join biotechnology and they can fulfill their dreams. In placement, there are 4 different domains. You can go for research and development or sales and marketing, or you can opt higher education or entrepreneurship.
Biotechnology has universal application in the field of biology. It has given heart impulse to the spinach leaf. Nowadays, we have all witnessed the application of biotechnology during this pandemic for drug designing, drug discovering and the vaccine designing. In industrial biotechnology, it has replaced the synthetic pigments by the fungus pigmentations to give the natural color to the food products. In industrial biotechnology, it has managed the waste products and enhanced its productivity by enzyme technology. Using the nanotechnology, it has used or designed the Botox injection to reduce our aging.
Amity Institute of Biotechnology provides students with wonderful opportunities to learn through practical approach. Here we are having multiple numbers of advanced biotechnological instruments which helps the students to learn the basic and the details, like we are having laminar, autoclave, distillation unit and many more.
At Amity Institute of Biotechnology, Amity University, Jharkhand, we have PCR, polymerase chain reaction instrument for the detection of molecular diseases, including COVID-19.
It's rightly said that books are the best friend and one can get it from library. Amity Institute of Biotechnology Library has number of books related to the courses we are running here. It includes textbooks, reference books, magazines and research journals. Most of the reputed international journals are online, available to faculty members as well as students by using unique login ID provided to them. For upcoming students, library might be a wonderful place to start their academic journey to get the degree in biotechnology.
Thank you, Amit sir. As rightly said, that education is the name of our attitude, actions, language, behavior, and personalities with other in real life. And science and biotech, as well as biotechnology, is a systematic enterprise that builds and organizes knowledge in form of testable explanation and prediction about the universe. Now, I request Dr. Amit Kumar Datta sir to kindly share a video showcasing a brief introduction about Amity University, Jharkhand. Over to Amit Kumar Datta sir.
Ranchi is part of India's leading not-for-profit education group, wherein over 175,000 students are pursuing world-class education across 10 universities, 26 schools, and 16 campuses worldwide. Amity University Ranchi has been awarded as the best private university of Jharkhand by ICCOB at a glittering ceremony by the Honorable Governor of Jharkhand. It has also been awarded as the best university for promoting industry-academia interface by ICCOB in 2019 and 2020. The university offers career-focused UG and PG degree programs across diverse disciplines from engineering to management, and from law to forestry to students from over 15 states besides neighboring nations like Nepal and Bangladesh. The students learn beyond the classroom at the state-of-the-art engineering and computer science labs, learning studios spanning mass comm and English language, and moot court for law, among others. Its fully automated central library houses thousands of books and online journals that can be accessed at a large number of workstations for in-depth research. Amity Ranchi's prestigious recognitions are a reflection of its diverse initiatives in high-end research by its faculty. who have filed several patents and published over 300 research papers in peer-reviewed journals, books, and book chapters. The faculty is also conducting several government-funded research projects for Department of Science and Technology, Government of India, UGC, Vigyana Prasar, GAIL, and National Board of Higher Mathematics. The Industry Interaction Placement Cell at Amity Ranchi is a student-driven body that acts as an interface between the students, faculty, and the corporate world. It maintains close contact with recruiters throughout the year and conducts numerous workshops and group discussions and mock interviews to prepare students for the corporate world. During the lockdown last year, Amity has organized over 100 webinars to facilitate students' interaction with Nobel laureates, industry leaders, and leading scientists. The university also has strategic tie-ups with leading institutions to facilitate skills enhancement workshops among students. Amity's overall focus on employability has also led to its students being offered campus placements in over 100 leading corporates last year. Amity University Ranchi is committed to emerge as a global education hub, nurturing the most brilliant students and attracting distinguished faculty who share our mission to change the world together.
Thank you, sir. Now, I would request the organizing secretary of the two-day international conference, Dr. Dhananjay Kumar Pandey, sir, to kindly propose the vote of thanks for the inaugural session. Over to Dr. Dhananjay Kumar Pandey, sir.
Thank you, Nimi madam. Hope I am audible.
Yes, sir, you are audible.
Okay. So it's my privilege to give the vote of thanks for this inaugural session, and with the motivations and blessings of Honorable Founder President Dr. Ashok Kumar Chauhan sir, our Honorable Chancellor Dr. Atul Chauhan sir, Honorable Senior Vice President Sri Ramchandran sir, Honorable Vice President Sri Gaurav Gupta sir, it was possible to conduct this international conference. We are very much thankful to our Honorable Vice Chancellor, Professor Dr. Raman Kumar Jha sir, respected Director, Professor Dr. Ajit Kumar Pandey sir, respected Registrar, Mr. Prabhakar Tripathi sir, for their continuous guidance, blessings, and supports during entire organizations of this conference. We are also very much thankful for our Deputy Director, Admissions and Marketing, Sri Sumit Singh sir, Assistant Director, Dr. Prabhat Tripathi sir, for their guidance and support. We are very much thankful to our Program Coordinator, Dr. Viswarup Samanta sir, our University Research and Development Coordinator, Dr. Jaita Chattopadhyay madam, for their guidance and support during this conference organization process. We are very much thankful to our distinguished guest, Dr. Aarti Sanwari madam and Dr. Kalavani madam today, to accept our invitation and for giving insights in challenges and opportunities in biotechnology during inaugural session. We are very much thankful to all keynote speakers and participants to accept our invitations and their active participation and sending their abstract and all things which made this conference successful. We are thankful to admin team of AUJ for their support during organization of this conference. We are also thankful to IT team Noida and AUJ team, IT team AUJ, Amity University Jharkhand, for their support and to host this international conference on Zoom platform. We are also thankful to Media Cell, Amity University Jharkhand, for their support and circulation of information regarding this conference widely. Last but not least, we are very much thankful to all participants to join this conference. And thank you all, welcome all, and thank you all. Now over to Dr. Nimi madam for further proceedings. Ma'am. Dr. Nimi ma'am. Am I audible?
Am I audible, sir?
Yes, yes, ma'am. You are audible. Over to you, ma'am.
Sorry for the interruption in between. 30 a.m. for the first technical session. And thank you all for joining.
Ma'am, I think you are muted. Few words we could not see. So could you please repeat what you announced? Because maybe some technical problems was there. That's why we could not listen. So for participants information, could you please repeat what next we are going to do?
Okay, so we will again resume at 11:30 a.m. for the first technical session.
Thank you.
And thank you all for joining.
Thank you so much.
So welcoming you all again to the day one first technical session. To start with, I would now welcome our first keynote speaker of the day, Dr. Kalaivani ma'am. But before that, before the start, here is a brief introduction about the academic and research profile of Dr. Kalaivani ma'am. Dr. Kalaivani ma'am has obtained her doctoral degree in area of molecular biology from International Center for Genetic Engineering and Biotechnology, JNE. Ma'am, research interest lies in area of diagnostic kit development for detection of causative organism for different ailments and drug development. She has handled more than 10 research projects granted by various funding agencies and has 100 plus publication in various national and international journals of repute. Currently, she is serving as the head, Department of Basic Science, the Open University, Sri Lanka. Welcome, ma'am, and over to you.
I'm audible and you can see the shared screen as well, isn't it? Can you all see the shared screen?
Yes, madam. We can see the.
And you can
to add notes. It's still not in PowerPoint mode. Now it's PowerPoint mode. Yeah.
Right?
Yes, ma'am.
Okay. Good afternoon, everybody. The Vice Chancellor, Registrar, the panelists, organizers, faculties, academics, participants, and students. I take this opportunity to thank the organizers, especially Dr. Amit Kumar, to inviting me as the chief guest to deliver the keynote address at the International Conference on Challenges and Opportunities in Biotechnology, organized by Amity Institute of Biotechnology, Amity University, Ranji, India. So I'm happy to share my knowledge in this special event at this moment. So let's move on to the presentation. So you all welcoming me through this virtual platform. So to communicate each other under this pandemic situation. So because if we travel through the I am reducing this one, like the if you travel through this and if it come to the place you and me, like so the question is, we are in the risk, so is it is a risk for everybody, and we have the fear as well. So that the current scenario, the pandemic, during this pandemic situation, it's emerging pathogen is coming up. The new microbial infections, which is a threat to the entire world. So it's not only to entire nation and to everybody in a risk. So thinking about this current scenario, the topic selected to talk here, a new era in modern biotechnology, challenges of pathogen discovery and prevention strategies. Let's move on to I can't move the slide. Okay, sorry for the interruptions. So we are happy, we are living happily. So the same time, how we are happy, the microorganisms also happy, which surround us, they are happy. So unless if we disturb the nature, so there won't be an issue that we happy, the microorganisms are happy. Like, but whatever the microorganism or the pathogen which comes, which encounters us, so then we have the immune system to protect from the pathogens. We are our immune system, the B cells, the plasma cells, B cell get activated due to the B cell activation, the antibody produced, and which clear the pathogen which comes to our body. But these are the pathogens, basically these are infectious, so biological agent. It may be virus, bacteria, fungus, or other organism. Those are the things in first link in the chain of infections and diseases. So, but what happened, like, unless the issue comes with if our immune system failure, or if there is a disorder in the immune system, then we need to approach the doctor for the treatment. But if the treatment, it may be possible. So what can be used as a remedy? Like modern vaccines are coming up, so it's there. And if it is especially for the viral infections, the vaccines production and for other organisms as well. And the antibiotics especially focus on this bacterial infection, and not only the treatment protocol, and also other prevention strategies, infection control measures, and so on. So if the treatment is there, what is the issue? The challenges, right? In case of the bacterial infection, there are growing rate of the bacterial resistant bacteria, multi-resistant bacteria. In the case of the viral infections, there are new variants are coming up. So that's continuously that changing due to the mutation, the variants which is coming up. So, but in the case of the bacterial infection, there are this major issue at currently the chronic biofilm-based infection, which are resistant to antibiotics. So what is biofilm? It is a group of bacteria consortium, that's accumulation of microorganism that when it attached to a surface, which irreversibly attached to a surface, and generally it's enclosed in a polysaccharide matrix. When it is attached to this surface, so it may form the matrix to protect from the environmental outside factors, and which live happily. So when it is detached from that, it can release as a planktonic form, either planktonic form, or it can be as a biofilm bacteria surrounded by the matrix. It is very common and it can be observed in any different places, even the washrooms and things. And but major thing that issues are in the hospital setups. This is a major issue with the medical devices. Among those medical devices, the indwelling urinary catheters, these are the major common sites of biofilm producing bacteria. This biofilm production in urinary catheters, which can cause the chronic infection in patients. If it is not treated, that leads to the death as well. So if that cannot be, if it is treated, then again, what is the issue? Issue with the treatment. Again, that is the issue is with the antibiotic resistance. These biofilm bacteria are very highly resistant to the antibiotics, and which leading the cause of therapeutic failure. Even the treatment is possible, but that due to this resistance nature, that there will be a failure in therapeutics. So there are many bacteria which has been identified as the resistant bacteria. Most commonly, the hospital-associated Staphylococcus aureus has been identified as a methicillin resistant, and even the multi-resistant to other antibiotics as well. So how this antibiotic resistance is developed? So this is a normal flora where it can have drug resistance germ. The bacteria may have a few, but what happened because of the nature, when we overuse of this antibiotic and the disposal of the antibiotics to the waste environment, when it is that make a difference to the normal flora. So that will kill not only the drug resistant, not only kill all the bacteria, so it will give kill the good bacteria and protect the infection from our body. That drug resistant bacteria remains. Even one bacteria which is remain, that's take over, and that will dominate in the flora. And even that bacteria, it is possible to transfer the gene to other bacteria, and that develop the resistance more and more, even with the treatment. So if you look at the gene here, the MEK gene, so that is the gene which are responsible for the this resistant and whether it is susceptible or resistant to the antibiotics. So basically this MEK gene that for the penicillin, and not only limited to that, so including the methicillin and other bacteria as well. So this is all related to B-lactam antibiotic family. So then when you look at the expression pattern, like when it is that is the penicillin binding protein PB2, that when the antibiotic, the methicillin, when it binds to the receptor that which cannot function, the receptor, then it comes resistant, it cannot function, then it becomes susceptible to bacteria. But in the case of the resistant, so where this conformation, the receptor conformation, which change the conformation, and when it binds to the bacteria, when it is bind to the antibiotic, so that cannot function as then this can involve in the cell wall synthesis of the bacteria and resulting loss of target affinity. So then that also depend on the expression of the MEK gene, which control the antibiotic resistance depending on the conformation which express whether it is the resistant or susceptible to the particular antibiotics. So these are the things commonly all the scientists does, very basic stuff. The specific medium used to screen, this is focus on the Staphylococcus aureus, and then Staphylococcus specific primers for the PCR reaction amplify to confirm the specific gene fragment, and also the MEK gene amplification and analysis by the bioinformatic tools, and identify the bacteria and the gene presence and the expression patterns, so on. But these are currently practiced everywhere. This is the current testing methods in the current biotechnology world. The molecular-based methods and serological and immunological-based testing methods are very commonly used, and all the scientists and all. Even in the COVID-19 pandemic, during the pandemic situation, the patients, the public have been tested for all these, using these tools for PCR, real-time PCR, and so on, and also the antigen test, the immunological based test, and so on. But it is very simple when it is in sample collected from the infected person, the clinical samples, where the targeted fragment is amplified, and the sequencing results which give the details of the sequence and which compare with the already established databases, and they identify the new variant we compared with the variant which has already been there in the database. So by which then that sequence information which lead to to develop the vaccine and so on. So, but what is needed? Like that is all the biotechnologists, all the current world having these tools and all in the hand, and so that is. But the question is, why this delay, and why this technology doesn't help us to immediately eradicate all these pathogens which is coming time to time with the new variants. So therefore the novel technologies is needed so to detect the pathogens. So why this? Because the research and development activities which need to aim to reduce the assay time. The time is very important factor, and to reduce the amount of manual labor by automating methods, and the sensitivity of assessment in cases where potential risk of infection, even the lower number, even a single pathogen may cause the infectious diseases. So that also, in a matter, we have to consider that for the future. So how this novel technology will help to move forward to reduce the time, labor, and the sensitiveness. So keeping that in the information, we are entering to this genomic era, pathogen detection. It's a great challenge to the public health and the clinical microbiologists. So because this, the time to time, the pathogen emerging and re-emerging pathogens which are coming up, and these are leads to the emergence of infectious diseases. And so this introducing the molecular diagnostic, so that is that will revolutionize the clinical microbiology, because we have the ability to detect even the minute amount of nucleic acid in the clinical sample, even a single pathogen. It maybe if it is in the clinical sample or from the sample which is collected from the infected person, so that it is a gift for the biotechnologists, molecular biologists to detect the pathogen. So therefore, we have to talk about the high throughput sequencing method, because these methods, so considering the basic, the molecular techniques, and why we need to think about more and high throughput sequencing method, because these billions of these nucleic acid fragments, which can be simultaneously and independently can be sequenced. So not as a single reaction or not the detection of single pathogen, it's a number of pathogen, it can be detected at one shot. Then rapid sequencing, the time that I mentioned earlier, rapid sequencing of suspected outbreak strains can inform both that will lead to the clinical practice as well as how the infection control procedures. And the level of genetic variation observed among strains, epidemiology is linked to an outbreak, which can help to determine whether it is a single pathogen or point that we analyze, or the multiple strains are responsive. You know, now the time to time, now we were talking about the alpha, beta, gamma, different variants are coming up. And so we don't need to go for single reaction time to time, everything whenever. But if we can move forward with this high throughput DNA sequencing at once, like we can do the sequence for the whole population from the infected samples. So these are culture independent samples, like you can use this high throughput DNA sequencing, enable large number of these metagenomic analysis in a system. That's the technology come with the next generation sequencing. These are also commonly adopted in most of the clinical laboratories. So what is the good thing is these are unbiased, not a specific protocol for detection of a single pathogen. So this is possible to identify even the co-infection and the multiple variant at once to there. And also there is no any newcomer may come, so anybody comes, so the technology is available. It's not a new it may be a new variant, or them again and again it merge one or whatever it is. Prior knowledge is not needed, but that can be possible through this next generation sequence. But it even this next generation not only restricted or limited to a infected sample or to the hospital sample, or even it can be applicable for any base sample, environmental sample. Even the DNA can be extracted directly from the base environment. And also here the focus, you can see these are the quarantine samples, not only then that also possible quarantine plant material. So that can be amplified if the bacterial infection, if it is focused, then it can go for the 16S RNA amplification. Where these the variable regions, so the interested variable region can be considered to amplify whatever the amplification here, using only focus with the bacterial genomes, the metagenomic DNA, but the pool of DNA from different organisms in the quarantine sample. So that possibly that will give the profile, the description of the pathogen, whatever the whole set of pathogens present in the sample. It may be in quarantine sample or the same protocol for any other samples you analyze. Here, then out of this, the focus can be here, the quarantine pathogenic organism, not the other non-pathogenic organism or things. So that can be identified and can be compared with other organisms, the place where it is placed, and the particular sequence can be analyzed. The same way it can be applicable for any pathogens from any source. It may be common for agriculture, medical field, or marine, or other bioinformatic field. Every thing it is common. So then this is the basic technique. It's not a different technique from the other basic molecular techniques, where it can be either isolate DNA or RNA. If it is RNA extraction, it have to go for reverse transcription and the genome amplification and next generation sequencing analysis. So if you look at, you are very familiar with this target specific RNA, real-time PCR, and also the metagenomic analysis, the NGS, and where it is possible here in the case of this real-time PCR, we are target specific PCR, but in the case of the NGS, we are focusing on the total genomic DNA. But the thing is here, when you look at the cost also, though the primary cost is more when you compare to this real-time PCR, where the analysis cost is more. But when you look at the output, like here you have to go one by one, and then you have to analyze a single reaction at a time. Then when you do analyze for the novel pathogens and many novel pathogens time to time, and that may cause the cost is more than this. But here, of course, when you look at one sample, there are multiple pathogen at a time. Even what next, the new variants coming up, everything that you can analyze at a one shot, and then the cost will be less. But this will be a slower time, but this will be the real-time PCR. It may be quicker compared to the particular reaction, but when it is overall output is a time and the cost and everything lesser than this, and even advantages, you will be able to go for the identification of new pathogens and the pathogen which is unknown. And when you move on to the next tool, the potential diagnostic tool is nowadays very, it's coming up, the aptamer. Aptamer is a single strand DNA which has the higher affinity with the target molecule. It may be single strand DNA or RNA, and which binds with this aptamer sequence. The primary sequence alone, it doesn't have much effect, but when it comes to the functional aptamer, where it's bind, fold it in a folder, it comes as a folding structure, or the bulk like structure, that is the 3D structure formation. It's become functional. So this can be this aptamer, functional aptamer, could be bind to any target cell, where it can bind to any biomarker. It may be a protein, it may be a cell, a molecule, nucleic acid, or any viral particle or anything. But if the conformations are compatible to each other, it can be. It is similar like the antibodies. Now this is called alternative antibodies, something which is replacing the antibodies, and which has the higher affinity and the specificity. The specificity which comes with this structure, this 3D structure of the aptamers. This affinity, where the interaction between these target and the aptamer molecule, van der Waals forces, and electrostatic interaction between charge groups and hydrogen bonds, which make the thing. So if you are focusing on the particular aptamer for a particular screening, then you however that go for with the random sequence, start with the random sequence. This is the random aptamer library, and when you incubate with the target molecule, whatever the having the higher affinity and the specificity which will bind together, and then that so the rest can be washed away, the unbound sequence can be removed, and then this cycle can be go again and again, maybe 5 to 20 times, to make to optimize as much as possible the binding of the target and the aptamer molecule. The unbound sequence can be removed, and then these bound sequences can be characterized either amplification or any other method, and then you can get the information, you can identify the pathogen or whatever particular molecule where this aptamer has been bound firmly, that can be detected by this systematic, it's called SELEX, systematic evolution of ligand by exponential enrichment. So this called by different name when it is focused on cell and non-cell select cycle. So the same procedure, you can see that here the random pool of polyconic bound to the target cell, and then again the same thing, it's repeating, and it can be amplified, and the sequence details you will be able to analyze. And usually these are created by selecting them from a larger random sequences, so and which has the potential in the diagnostic, especially that will be now currently it is highly recognized, and then that is recommended as well to go for these pathogens detection. And interesting things is not only to for the diagnostic purpose, but also it is commonly used in therapeutics as well. So as I mentioned earlier, this biofilm bacteria, where the bacteria which attach to the surface and which form a matrix and which enclosed in the polysaccharide, that is the biofilm maturation when it is released, and that's planktonic bacteria, or even the planktonic bacteria which it comes, and when it is attached to the surface, that becomes the biofilm bacteria. So what's happened when we during the therapeutics, that applying this aptamer, and which binds to the planktonic bacteria and which prevent the binding to the surface, that inhibit the biofilm formation of this. So that is possible not only to diagnose it, but also it's possible to go for the therapeutics as well. So then let's jump to the nanotechnology, where so the molecular, even in the biotechnology, where the molecular biology alone do things, not alone to do the things, and also join with the nanotechnology, where so as now the scientists, a lot of scientists working on these nanoparticles, different particles, and there are promising effect by the silver nanoparticles, and which has the strong antibacterial activity and so on. So they are trying to like coat the material with the to any surface. So if it is here we were talking about the urinary catheters, it is like that's for example like that. There are several other places where if we want to get rid of the antibacterial activity, that can be coated with nanoparticles. And here in this case, where the Staphylococcus is mostly identified as a biofilm producing bacteria, but there are certain cases it's non-biofilm bacteria. Also, it's a passive bacteria along with other bacteria sits together. It is present, that also in the from the experiment it has been proved and all. So then when it is coated, like it's prevent to attach with the surface. So that is an advantage for using this technology. But this is not starting from now. This is 1930s to up to now. That's leading Foley catheter technology over these more than 100 years. It is they try to coat it with several material to prevent this bacterial infection, to stop that chronic infections for future things. So they have coated with several silicone and elastomer and hydrophilic nature, so many things. Now even the people are working on these nanoparticles, and even the medicinal plants, secondary metabolites, to cover the places where they wanted to stop the antibacterial activity, and the research on the way. So what I would like to share with you all with this information on all the success story from Sri Lanka. So there's a nano product that Sri Lankan scientists introduced, a face mask. They developed this face mask with the materials improved using these technological techniques. So the research team from University of Peradeniya. I am from Open University of Sri Lanka. This is a research team from the University of Peradeniya. So they have released the nano product. So basically the face mask covered with this embedded with the nanoparticles. So the names released here: Respiron Nano AV99, Respiron Nano AV99. Now this is washable, protective and comfortable, and it can be reused, and the scientists, they have mentioned that it can be washable even 20 times, and there are different colors as well. These are environmentally friendly product, and so it consists of 3 layers, this innovative product. The outer layer is the superhydrobic cotton layer that repels aerosol particles, including droplets that can carry viral particles, and the middle layer, they have modified the cotton layer with the antiviral, antibacterial and mechanical properties, and the filter air using the electrostatic filtration. And the third and the innermost layer, they have produced as the hydrophilic layer that can absorb and evaporate the liquid droplets. So I just like, it's a 2 minutes, the video, I can share that they have released this video that is available in the net as well. So this is the
I think video is fine, ma'am. I think video is fine, but audio is not coming. Maybe some technical problem.
Hello, Kalaivani ma'am.
Yeah.
Ma'am, is there any audio in that particular video?
Yeah, you can't hear.
But ma'am, there is no sound is coming.
Sound is not coming.
Yes, ma'am.
I think we have to adjust share sound.
You just stop sharing, ma'am.
Now
after that, you just share the screen one.
Particles are used to partially block the pores of the woven cotton fabric in a mechanical bonding to a level of 300 nanometers, resulting in nanometers.
But now you can hear, no?
Yeah, ma'am. Now it is coming. You can start from the beginning. It will be very good, ma'am.
Respiron AV99 is an environmental friendly, biodegradable, low cost, reusable and antimicrobial face mask with water and blood repellency and stain resistance. It also repels and destroys any aerosolized viruses or bacteria. In this multifunctional face mask, the target has been to reduce both the waste burden and the impact on the environment. As such, cotton, a commonly available, natural degradable fabric material, has been used in 3 layers, nanotechnologically modified individually, to have unique inherent properties. The outer layer cotton fabric is super hydrophobic to repel any aerosolized viruses and microbes as well as blood or any other water-based stains, comparable to a lotus leaf shedding water. Any microbes penetrating the outer layer will reach the nanotechnologically modified middle layer with multimodal antimicrobial activity. Micro and nanoparticles are used to partially block the pores of the woven cotton fabric via chemical bonding to a level of 300 nanometers, resulting in nanometric range filtration. The adjustable ear loops ensure tight fitting to the face of the wearer. A unique mask design and a metal or plastic noseplate addresses the problem of air escape at the nasal bridge. The innermost layer is made up of 100% hydrophilic cotton to absorb carbon dioxide and moisture present in the exhaled air. This face mask can be reused even after 25 cycles of washing or disinfection, at least for a period of 1 month. The nanoparticles used in these masks are safe in a wide range of applications, including medical and cosmetic industry. High airflow tests and wash cycles have proven that the chemically bonded nanoparticles are stable and do not detach from the fabric surfaces. The mask has medical grade characteristics with high resistance to flammability, satisfactory results in differential pressure tests for breathability, and is highly resistant to blood and fluid permeation.
Okay, with this video, then we have to so what we can see that not only the diagnostic method, but also the preventive measures also important for to moving in this current world. So that is coming as a technological change. It's not a molecular biology, it's nanotechnology together. Then molecular nanotechnology will serve our purpose. So then the future is going to be smart. So the pathogens are smart. So why not we? So we have to put together the advanced technology and the innovation. So that would come together, and then there will be a solution or remedy for the smart future. It's in the future, we have to think about out of the box. So not only the technology and the lab confined to lab practicals and lab research or innovation and things like that, but little bit we have to think in the future, because currently we have faced during this lockdown period and the pandemic period. So there is no way the hospitals have full there are no seats to go and to detect these pathogens. So something we have to come up with some ideas. So how we can do this with the home made products. So creative home ideas also with the collaboration with the in between the countries, so in between the organization, in between the people and things in the universities, to come up with some idea what we can develop. That is something that has to need to develop at the home level. So that will transform the future to deal with the smart future, and not only with the future, and which create the new market force like commercialized product to fulfill the needs of the public. So this transforming future with from our knowledge to the technology to the commercialized product, that will help for our future generations. So we should accept the technology. Here, look at this cartoon. The guy was carrying the load, but there is no wheel, but suffering. But when the person is offering the wheel, even that person was saying, "No thanks, we are too busy." Like, we need to accept the technology. We need to move forward with technology for the future market. So then the biotechnology not only restricted to, as I said, the organization and universities or whatever it is like, but it has to, it is merged with the multidisciplinary things. So we need to collaborate with the different disciplinary people, that it's the different field, and even the collaboration should come in between the countries and so on. So with that message, to take over the to have a challenge, to face this challenge to the pandemic, and for the future, we don't know, it's not predicted. So then we have to be ready for the future, ready for the smart future, and all we scientists together, we have to be ready for to anything. And with that message, I would like to thank you very much, and thanks for the opportunities given by this Amity University and the organizers and everybody. Thanks. Have a nice day.
Thank you, Kala. Thank you, Kalavani ma'am, for sharing your valuable research experience in the field of pathogen discovery and prevention strategy. It was indeed a very informative session. Now, I would request our participant that if they have any query, then they can post their queries in the chat box section. So one query I have already received from one of our participants, that she wants to know the name of the scientist who have created the Respiron Nano AV99 face mask.
Yeah, that's in the video as well. Professor Rajabaksha.
Ma'am, kindly unmute yourself.
No, it's mute only, the unmute only. Can you hear me?
Yes, yes, ma'am.
Yeah, Professor Rajabaksha and his team, they are multidisciplinary team. They joined together and they have done this innovation. Can you hear me?
Yes, ma'am, you are audible.
Yeah, the Professor Rajabaksha from University of Peradeniya. So they have actually they have all these informations are available in the web. So I have collected from web, but luckily I had an opportunity also to listen his talk recently in other forum which happened in Sri Lanka very recently, 2 weeks back. So he has presented this work at this forum also. But when I was thinking to collect some information from him, and by the time I checked, that's available in even this video and thing, it's available in the net. You all can download and you can get this video and other information as well.
Thank you, ma'am. Anyone else having any query?
And yeah, it was audio that you were able to hear, no? The whole video, the sound also?
Yes, ma'am.
All right.
I request the participant to please ask their queries.
Meanwhile, may I ask one query to ma'am?
Yeah, sure.
Yes, sir, please.
Yeah, so actually, we have seen that the testing system for pathogens in your slides. So I have just one doubt that NGS is now
pathogens? I didn't get it. What?
Any pathogens? Any pathogens? For example, nowadays COVID is very common. So I have just one query that is the sensitivity or the accuracy of NGS is equivalent to RT-PCR nowadays we are doing, or there is some we miss?
Yeah, all the things, you know, these all the methods which are pros and cons, right? So, but if the RT-PCR failed to identify some pathogen, so it is better to go for the NGS. But basically these are the, you know, it has to work with the public. So it has to immediate need has to be fulfilled. So this NGS is not it's not commonly used techniques. It has to go with little bit sophisticated instrument at higher level in the laboratories. So, and the cost for just to check the particular pathogen to analyze the whole sample is not, it's not feasible. But in the future, right? So if but because we have already facing this pandemic situation, and at this level, it is better and it is recommended to do this analysis in the laboratory level and to see whether any other co-infection, any other new variants comes. But it's not advisable to do for a single person by using this method. This is as a common. So before getting into the pandemic and to have idea and about the new variants and any mixture of population in this particular sample, it is advisable to do the things and get the idea. So before the new variants say coming, then before it causes the infectious diseases, so it's better to analyze. But not after coming that it has to go for this real-time PCR or whatever the normal PCR.
Yes, ma'am. Thank you, ma'am. Thank you, ma'am.
I asked the
yes, ma'am.
Good answer, right? Yeah.
Thank you, ma'am. One of our participants would like to know that what is the current scenario of COVID-19 in Sri Lanka?
Yeah, now it's coming down because compared to like 3-4 weeks, but now it's back to normal. Now we all started our work. Every day we are going, and all the even the travel restrictions have been removed. And it's back to normal. But the data that the death and the infected person the records also not coming much because it's look like normal. But there are cases reported, but compared to 3 weeks back, so it is less.
Okay, ma'am. One of our students would like to know that what are your thoughts on chloroquine and hydroxychloroquine?
Yeah, actually, I can't because as like you and me and the public, this message is same for me as well. Because unless if we do the research on that, and as a scientist, we shouldn't tell our opinion. That's my belief. But we need to like these are the because these all I think it has to better if any consultant or any medical person, if they answer this will be a answer for that question. So maybe that we can anything we can tell about our thing if we prove that. So not the drugs that we introduce to the public.
Okay, ma'am. One more question I have received that what kind of nanoparticles are being used in Respiron Nano AV99 face mask?
So they have used the zinc oxide and other thing, and even the titanium oxides, that thing, when I listen that talk, they have mentioned that. But we have questioned that what about the silver nanoparticle, and because that has some toxicity issues and all. But however, when it is not in the form, like when it comes as a nanoparticle form, that the toxicity is not an issue. So that's what we question that, but they haven't used that the silver nanoparticle, but they have used the zinc nanoparticles, and they have tested for the public health as well, and there is no issue with that. So after that only they have released.
Thank you, ma'am, for answering the queries.
Yeah. Thank you for your,
any other participant who would has any queries, please post your query in the chat section.
Excuse me, ma'am. Ma'am, I am audible?
Yeah, sure.
Yeah.
Go ahead.
Ma'am, actually, there are 3 vaccines basically now in India we are basically using. That one is Covaxin, another one is Covishield, another third one is the Sputnik. What is your opinion? Why which one is the best? If we are taking Covishield, it will give more.
Can you please repeat? I didn't.
Ma'am.
But you are India?
Yes, ma'am. Now audible?
Yeah, sure. Go.
I mean, India, basically there are three types of vaccine name. The one is Covishield, another one is Covaxin, and the third one is the Sputnik. It's maximally widely used as a precautionary measure for the COVID-19. So someone is telling that if I am taking Covishield, it will be more good and more prominent, and someone is telling that Sputnik is better than Covishield. What is your opinion, ma'am, on that? If we are taking Sputnik, so it will be good that in future we will not be affected or attacked by further for corona. So what is your suggestion, ma'am?
Yeah, this all again that how they have made the vaccine and on what basis. And like as an antigen, so the epitopes, it can be changed. It can be changed, and also the time to time different. So as it is coming, it is in the paper and the news. So different variants are coming, different epitopes. Even if we produce the vaccine, it's not targeting for the particular epitope. So it may be, but it may fix to the other variant as well. But it's not completely. It is completely. We can't say that it will be recovered. So we have developed the immune to the particles. So like that. So we have to look at these different vaccines, how they have made, and we don't have much idea about the protocol and what really the vaccine targeting. So maybe there are the common region they might have focused, but still there may be difference. So that we can't answer at this moment. So with the time, with the emerging pathogen when it comes, so then that has to be give an answer for us. But unless if we do research and if we compare the sequence, what they have used, what the vaccine contain, then only we can, as a scientist, we can release our opinion. Thank you.
Thank you, ma'am. One of our participants as well as delegates asked Ms. Ketki Galani asked that what are your thoughts on herd immunity of this pandemic? And also he asked that how much time will it take to reach?
Question is what
are your thoughts on herd immunity?
Higher immunity.
Herd immunity, herd, H-E-R-D, herd immunity of this pandemic.
Immunity to the pandemic. So then we have to develop. I don't know whether I have understood the question correctly. So that means how we have developed the immunity and to tackle with this pandemic situation.
Yes, ma'am. Yes.
So then because we have to that's why the people say that different age people they all already have some transplantation or something. Then their immunity system goes down with the elderly people and the system if they have any other diseases and diabetic and things like that, their immunity is lesser than compared to other people. So then this pathogen can easily encounter, and that may have some impact on their body health. But if we can boost our immunity nowadays, that's why the people are advertising like different medicinal plants and different these all spices and all. So that will all the other vitamins and things like that. If we boost our immune system, so we are like whatever, because we have lived with these microbes, for from the time that the human and the other animals created here until such time. So there are the pathogens which are surrounded by us. We are also living. That's why at the initial talk I told me happily we are here. But now the system, now the nature, now the environmental, which we have polluted, which we have made the changes, that the activities of human, which has the impact on the environment, the natural balance, the equilibrium, which has gone away, which has disturbed. So the nature itself, that's why I said the overuse of antibiotics and throwing antibiotics, the hospital waste and all. So that disturb the system and the microorganism. That's why this emerging microorganism, which that also has to live, that also like the organism like us, that also has to be in the world. It has to be there. So that we disturb the system and our technology moving advanced, and we have created so many things. Then the pathogen. Now we have to go in another way, reverse way, and then what only thing that we have to go back to the basic stuff. The basic stuff, what is boost our immune system. So then whoever comes, like then we are strong. Like a military situation, the terrorist is attacking the boundary. So even in the security at the at the point. So they are easily, if there is no war or there is no barriers, then everybody is moving to through the gate and all. There is no restriction. But when something happen, then they all alert. So they all stop each other and asking questions, things like that. So if we are firm, if we have developed our immune system in a better way, we have boosted the system. So whoever comes, whoever the pathogens comes, we don't need to worry about. So then we need to find a way. In addition to these technological advances, also we have to think about the system, how we have to identify the medicinal plant and the spices and things, which we have to do some research on that area as well. So how we can boost our immune system. So then easy to both applying the boosting system and to considering all this technological way. It's not a traditional way. It's the medicinal plants. Also now we are going isolating secondary metabolites and these compounds. These go for the treatments. So that all will help. So putting together, that's why I said this technological world, it's a chain. So putting not one, so together, that will make a success. That will make a difference. So then it comes with the innovation.
Thank you, ma'am, for providing such valuable information. And also thank you for accepting our invite for this online conference. And very soon we are accepting that you will accept our invite for offline conference soon as the pandemic is over. So thank you.
Sure, thank you. I have been in India several times. So I came to talk also to 3 talks in India, even through virtual also in India. This May also I had another talk. So I really enjoying. So I like to share my knowledge and the experience. I'm not that expert, but whatever the thoughts, whatever we do and whatever that can be shared with you all. So it's a great opportunity also. I'm thankful for all of you all and nice meeting with you all also, even though through virtual platform. Thank you very much.
Thank you so much, ma'am. Now, I would like to extend my warm welcome to the second keynote speaker of the day, Dr. Piotr, who has joined us for today's event. Before asking him to share his research experience and to enlighten the audience with his fountain of knowledge, here is a brief profile of Dr. Piotr. Dr. Piotr is currently working as an associate professor at Adam Mickiewicz University, Poland. From 2011 to 2015, Sir has served the prestigious University of Cambridge as postdoctoral research associate. Actively doing research in the area of plant genetics, Sir has published various research and review articles in high impact factor journal and also has one international patent granted under his name. So welcome, sir, and over to you to continue for the session.
Okay. Welcome everybody. I just will start sharing my screen. Is it visible?
Yes, sir, it's visible.
Okay, so Yeah, first I would like to thank the organizers for the opportunity to be on this conference and present our research. I will be talking about crossover control in plants, about the process of recombination that is happening during production of gametes, and I will make, sorry? Is something wrong?
No, it's fine. Please go ahead.
Okay,
It's just background noises.
Is the
It's fine.
presentation? It's fine. Okay.
Yeah.
All right. So yeah, I will be talking a little bit about how we can use the factors that are involved in crossover control for developing new breeding strategies and why it is so important. Okay. So of course, first I need to give you some more general introduction. So normally living organisms, eukaryotes, they divide themselves using a process that is called mitosis. And in mitosis, we start with one cell that has two sets of chromosomes coming from both parents, from mother and father. And then during this process, the cell is divided and produces two descendant cells that also has the same information, because during the mitosis process, the whole genome, the whole set of chromosomes is duplicated. So at the end, from one mitotic cell, mitosis cells, from one somatic cell, we produce two cells that are identical to the parental cell. And this is actually the way all the cells in our bodies and also in plants, in animals, divide by a regular way, and this is how we build our bodies and how we can develop and grow. But also there is another type of cell division that is restricted only to very specific process, that is production of gametes for sexual reproduction. And this process is called meiosis. It also starts from diploid cells, so cells that can have chromosomes from mother and father. However, during this process, two successive cell division happens. And because of that, the number of chromosomes is reduced by half at the end of the meiosis. So as you can see on the scheme, we start from 2n, so where n is a number of chromosomes, but end with 1n, showing that the number of chromosomes is reduced. And what is the most important is that during this process, the information from both parents need to be recombined in a process that is called crossover. So crossover, so during meiosis, chromosomes from father and mother meet, pair, and then exchange their fragments in this crossover process. So at the end of recombination, you have chromosomes that has part being from mother and then another part being from father. And this is actually called crossover recombination. So because this happens in all meiotic cells during the cell meiosis, then the final products of meiosis are very different from the ancestor cell, because the information is recombined between the genetic information that comes from both parents. And this is actually the major reason of variation that we observe in natural populations. We also, this is also the reason why children are so different from their parents, because the material that is inherited from mother and father is recombined by crossover. So yeah, just to sum up, crossover enables shuffling of genetic material, and this is that comes from mother and father, and this is one of the major importance of this process. But there is another very important reason why during meiosis crossover are formed, and it is to ensure proper chromosome segregation. So if we don't have crossover between a pair of chromosomes, we can end up with missegregation of chromosomes that can cause many different problems and abnormalities. For instance, Down syndrome, that is caused by trisomy of chromosome 21 in human, is usually caused by the lack of crossover between the two chromosomes 21 coming from mother and father. So now when you know a little bit about more general things about crossover, the question comes when, where, and how frequently crossover happens in the cell, and what factors determine crossover formation. So one thing that we also need to know about crossover is that the number of crossover is very tightly controlled. There are many different levels at which this process is controlled, but the most important things is so-called obligatory crossover and crossover homeostasis, two processes that control crossover. Obligatory crossover is a phenomenon in which ensure that each pair of chromosomes, so pair coming of two chromosomes coming, one from mother and one from father, each of them need to get at least one crossover per meiosis. This is to make sure that they will properly segregate in the later stages of cell division. The crossover homeostasis is a process that keeps crossover number at a very stable level, independently on the situation, external situations, external conditions. And now I want you to take a look on this graph showing crossover in different species, in different eukaryotic species. So here on the y-axis is the average crossover number per chromosome pair, and each data point here represent one chromosome, and color and shape of the data points indicate the species. So for instance, you see this data points in gray here, and this corresponds to budding yeast chromosomes. Here are this squares, blue squares corresponds to human chromosomes, and here in green you see chromosomes in barley. So what is interesting is that independently on genome size, the number of crossover is always adjusted to the shortest chromosome and is kept at a very low level. So for instance, the shortest chromosome of budding yeast is just half megabase pair. This is the physical length of the chromosome, half megabase pair, and it achieves normally about 2 crossovers. But in barley, the shortest chromosome is almost 1,000 megabase pair. So it is about 20,000 times bigger, but it still achieved only 2 or 3 crossovers. So it shows that
Professor Piotr, sorry for interruption, because now we are visualizing more than one slides together, means other slides also. So can we keep this?
All right. So I will try to maybe stop presentation for maybe I will just start and then share. This is not improved, yeah?
Yeah.
Is it better now?
Still.
Okay. Yeah, sorry for this technical issue.
No problems. No problem. I think we can keep it in PowerPoint mode, if possible.
Maybe I will first start. No, it's impossible, I think. Sorry, I will just try once more.
Yeah, I think this is better. This is fine. Now it's fine.
Okay. Yeah, sorry. I
problem.
wasn't aware of this.
No problem.
Okay, so yeah, so coming back to this slide, so you can see that for some reason the crossover number in all organisms is kept at a very low level. Whatever species you will take here, you will see that the shortest chromosome, the crossover number is adjusted to the shortest chromosome, and it achieved about 1 or 2 crossover per the genome. And also importantly, the crossover distribution along the chromosome is not even. So this is an example of Barley chromosome 7, and as you can, and here the red line represents crossover frequency. So you can see that there is a lot of crossovers at the very end of chromosomes of the chromosome, but there's not much inside. So there is nothing going on inside the chromosome. And because of that, in many species, in many plant species, only the end of chromosomes can efficiently recombine, while the rest of the genome is not really released for recombination. And importantly, there are very frequently important genes in the region that is not able to recombine. I will show you in a moment why it is important. So actually, meiotic recombination, crossover recombination, is the major tool used by breeders to combine different genes, different alleles from different varieties. Let's imagine that we've got a plant that is resistant to some viruses, or for instance, it could be resistant to drought stress or to some pests, and another variety that produces high yield, shows high yield. So of course, breeders want to bring together those two features, those two traits, to have the variety, the cultivar, that will be both resistant and also will produce a lot of seed or other things that will be used as food. So to do this, we need to the breeder needs to cross those two lines, those two plants, and then wait until recombination will transfer the beneficial alleles, beneficial genes from one variety to another variety to build this recombined plant. But of course, breeder does it without knowing about the recombination, and because of that, it takes a lot of time, especially in situations where genes that are responsible for this resistance are located close to the region that are not really combining. So because of that, the process of generation of new varieties can take a lot of time and is not very efficient. We can also of course use GMO, but in many cases we don't know which gene to introduce to for instance increase high yield or resistance, because it's very common that we don't really know we don't know what is the gene that is encoding resistant to some pathogens, to some fungi or climate, high temperatures, for instance. So the other thing that is very important to remember is that at the moment that the plant breeding is crucial for food security in the world. So when we plot the human population, we will notice that it is growing, and by 2050 it will almost reach 10 billions. At the same time, the cultivation area that we use to produce food is not changing, because our planet is still the same. So we cannot increase significantly the amount of land that we can use for agricultural production. So when you calculate the cultivation area per capita, so per one person, you will see that with time we have less and less area per each individual, per each person. And because of that, this area needs to be more efficiently used to produce food. Otherwise, we will not be able to produce enough food to fulfill the requirements of the growing human population. So that's why increasing recombination can speed up the breeding process. And by this way, it can help to secure food for the growing population. And I'm looking for factors that can boost recombination in plants. However, for many different reasons, I'm not using, I'm using a little bit, but I'm mostly working on the model organism, Arabidopsis. This is a very small plant that is not used for food production, but it is very suitable for different type of laboratory experiment. It is very small, you can grow it very easily, and it has very short life cycle. So because of that, it could be very nice model for studying plant genetics. And also importantly, it's closely related to, for instance, a brassica like rapeseed, cabbages, cauliflower, broccoli, and other species from this region, from these families. It also shows a huge natural variation. You can see here different Arabidopsis plants from different ecotypes, from different accessions. They differ significantly, and because of that, they are a good source of different variation that we can further study. So, to measure recombination, I told you that it is quite difficult to study recombination because the number of crossover is very low. So because of that, we developed a system that allows us to very efficiently measure recombination, and it is based on fluorescent markers. So we've got a line in which on one chromosome we've got two genes expressing fluorescent reporter protein, red fluorescent and green fluorescent. They are on the same chromosome, close to each other. And when we cross this kind of plant that has these two reporters on one chromosome with another accession of Arabidopsis, then crossover can happen within this region. And if crossover happen here, we will observe that as a product of meiosis, we will get chromosome that has only one fluorescent reporter, red or green. So these are recombinant chromosomes, but most of the chromosomes will be non-recombinant, so we'll have both reporters or non-reporter. And importantly, those reporters are expressed only in seed. So we can collect our seeds from Arabidopsis. This is Arabidopsis seed under the microscope. You can see each plant can produce a few thousand of seeds. And then after the fluorescent lamp, you will see that those seeds show different pattern of fluorescence. So red seed and green seed corresponds to those recombinant seed. And yellow seed and non-color seed that we can also spot here corresponds to parental combination that did not came through recombination in this region determined by the two reporters. And then we can easily calculate number of recombinant versus non-recombinant seed, and based on this estimate what is the recombination frequency in the region that is defined by the two reporters. So we use the automatic seed scoring for this that they developed several years ago. Okay, and now having the very efficient method to measure recombination, we can use some approach to perform QTL mapping. So QTL mapping is a strategy in which we want to associate particular trait with a genotype, with a gene that are encoding this trait. And to do so, we crossed the line that is the Arabidopsis line that has these two reporters on chromosome 3. You can see Arabidopsis has five chromosomes, and the reporter is here. So we cross this line with another accession of Arabidopsis. We excluded, shown here in red, one chromosome was excluded from the analysis because this is the chromosome which we used for measuring the recombination. So to not have additional effects, we excluded this from analysis. And then in F1, you get a situation in which we've got one chromosome from one parent and one chromosome from another parent. So this is heterozygous state. But in the next generation, when we self-fertilize the plant, then each plant will have completely different pattern of genetic information. You can see here this mosaic of segments. So there are segments that has fragments from mother and father, but also there are segments on chromosome that has only information from one parent. Yeah, in this case, for instance, from Ler, from this accession Ler, and another accession Col. Yeah, so you can see this pattern, and each individual will have a little bit different pattern. And then for each individual that we will get in this population, we can measure recombination using the automatic seed scoring system and genotype the plant. So check what is the structure of this genome, and then try to associate those two factors. And by doing this, we can then plot association between recombination and the genotype. And this is this kind of plot. You can see here 4 chromosomes that we use for mapping. So this is the location on the chromosome, coordinate on the chromosome, and markers are also shown. And here is a statistical test that is showing you whether some region is strongly associated with crossover control. So you can see here that we found in this population, we found 2 QTLs, 2 loci that are strongly associated with control with recombination frequency, one on chromosome 1 and another one on chromosome 4. Interestingly, you can also see how this association looks like. So here on this plot, we've got recombination frequency. So, and each dot here, each circle here represents one plant from this population. It was quite huge population of few hundreds individuals. So each plant was measured for recombination. And as you can see, they differ in recombination frequency. However, if in this region, QTL1, that we called QTL1, the plant has genetic information from only one parent, from this blue parent, col, so it was col. It shows a relatively high recombination frequency, while if this region was from the other parent, from Ler, so red one, then it shows low recombination frequency. This shows you that there is a strong association between those two features, recombination frequency and the genotype in this very region of the chromosome. And the same we could do for the other QTL. And then we performed a very extensive pattern mapping to find exactly what is the gene responsible for this feature. We used some other populations. Some of them were smaller, but in this case, we were just studying chromosome 1. We excluded all other chromosomes because we wanted just map the factor that is located on chromosome 1. And here we generated very big F3 population of more than 3,000 individuals. And based on markers, genetic markers, we could very precisely map where this gene is located, that it's connected with recombination frequency. You can see here this very huge plot of LOD50. Here are locations of markers that we use to count the statistical significance. And we could define by statistical ways, we could define the so-called credible interval. So interval of the chromosome that for sure is carrying this gene that is controlling recombination. And by this mean, we found this region between the two red lines. This is a credible interval that has only few genes, as you can see, several genes, but the peaking interval, so the interval between the two highest markers, has only 2 overlapping genes, this one and this one. These are two versions of this gene. And we know, we later found that this gene, this is HI10. HI10 was a gene that was previously described as the gene that has some role in meiotic recombination, is interacting with proteins that are doing meiotic recombination in plants. In many actually organisms, because High 10 is also conserved in human. In most of eukaryotes, we find homologs of this gene. Very interestingly, I cloned this gene. I cloned genetic fragment of this gene and transformed plants that were able to, in such a way that I could still measure recombination frequency. And this is the result of this experiment. Each dot here represents measurement of recombination frequency from one individual. So you can see that this is the control. It shows about 20 centimorgans. This is the recombination frequency in centimorgans. And then when we transformed with empty vector, the recombination was very similar, about 20 centimorgans. However, when we transformed the plants with genomic fragments encoding high 10 in different combinations coming from both LER or col parent, we always observe a very significant increase in the recombination frequency that you can see here. Yeah, the dots, each plant shows higher, most of the plants show much higher recombination frequency than the control. And moreover, we could isolate RNA and check how it correlates with the expression level of high 10. And we found that when this gene is highly expressed, the recombination frequency is higher. So it shows that actually high 10 is a crossover limiting factor in Arabidopsis. So the higher is the high 10 expression, the higher will be the recombination frequency. We also confirmed this by some cytological approaches. So here we can, for instance, count crossover. You can see that in wild type plants, you normally see 1 or 2 crossovers per chromosome pair. But in high 10, you can see up to 4 crossover per chromosome pair. We also used some other markers of crossover to do it cytologically and also observe this significant increase in crossover frequency. And later we wanted to see, because you need to remember that this system for measuring recombination is allowing us to measure only in this one interval, to measure recombination only on this one interval of the chromosome. So we wanted to see what is going on in the genome-wide scale. And for this, we used a recently developed technique for reconstruction of genome after INF2 population. So we took the line that is, we take the line that has this high 10 overexpressor, this additional copy of high 10 that boosts recombination, and cross it with another accession with LER. And then, of course, we got F1 plant and INF2 plant. Each plant will have, again, this pattern of different genome. This is not shown here, but each individual will have this different pattern of fragments from from one and another parent. And we can, based on this, estimate where crossover happened, because we can identify the regions from both parents. And this is actually shown on this movie here. So you can see here 5 Arabidopsis chromosomes, and the circle denotes centromeres of chromosomes. And anytime this color changes, it shows you another individual and the pattern of genomic fragments from one or the other parent. In green, as shown here, in blue is from one parent, in red is from another parent, and AB is a heterozygous, so it has fragments from, at the same time, fragments from both individuals. So you can see how we made this genome-wide. And when we calculated this recombination frequency from 200 individuals, then we could plot in the genome-wide scale the crossover distribution. And this is actually this plot. I'm just showing the average distribution over the chromosome arm, starting from telomeres to centromeres. So this is the data coming from all 200 individuals and from all 5 chromosomes. You can see that for wild type, this is the distribution of crossover. You see there is more crossover at the end of chromosome, but there's still a lot of crossover inside. And the recombination frequency for high 10 overexpressor for the line that has additional high 10 copies is significant increase, about twice 2 times increase along the chromosome. Also, these plots, these are histograms showing the number of crossover per individual. You can see also that normally it has Arabidopsis have about 7 crossovers, but in the line that is overexpressing high 10, you can see more than 15 crossovers per plant. So we significantly increase crossover frequency. And we patented this for Arabidopsis and also for maize. And at the moment, there are some groups, I'm not very involved in this because I was mostly working on this model organism, but now it's time to use this information to generate lines of plants that are important for food production, like rapeseed, wheat, also barley. These are the plants the crops that I know that people are trying to use high 10 to boost recombination and to speed up the breeding process. All right. Moreover, we also found that we can combine different factors that stimulate recombination to very significantly boost recombination. So another group described another gene, RECQ4, that is encoding the helicase, DNA helicase, and the mutant of those genes show increased recombination frequency. So we combined high 10 with this mutant and achieved a plant that is overexpressing high 10, so producing more high 10 protein, but also has mutated RECQ4 gene. And you can see that the recombination frequency was very significantly increased from 7 that we observed in wild type to 30 that we observed in this plant having both genes misregulated. And this is also the distribution of crossover along the chromosome arm. You can see that it significantly increases mostly in the chromosome arm. There's not much increase in pericentromeric regions, unfortunately. All right. So we've got still some time, I think. Yeah, I've got still some time, but I will maybe because it's like already 8:35 and we need to finish at 8. Well, in Poland it's 8:35 a.m. So we need to finish in 5 minutes. So maybe I will skip the second part that is showing how we identified the second QTL. This is located on chromosome 4. So we did a very similar approach. We also did all this large population, identified the region that is for sure encoding the gene, and then tested different mutants to identify the gene that is responsible for changing recombination frequency. We also did the complementation test to prove that this is the gene, and then the genome-wide distribution of crossover were identified and mapped. And also but unfortunately, the cytological characterization of this other gene that we found shows that the mutant of the gene that is increasing recombination frequency has some problems during meiosis. It's causing some chromosome fragmentation that you can observe here, chromosome fragments during meiosis. And of course, it makes some problems with fertility of this plant. And because of that, we think that this particular gene that we identified to be component of SMT56 complex cannot be used for plant breeding approaches. But just to summarize, I would just tell you that I think that I convinced you that increasing crossover frequency would significantly accelerate crop breeding for food production. That is very important in the conditions of changing climate and growing human population on the world. And also, I showed you that high 10 is a natural crossover limiting factor in plants. So when we increase high 10 production, then we can also significantly increase recombination, crossover recombination that could be beneficial in plant breeding. And also, so I showed that high 10 boosts recombination, and also that it is possible to combine different crossover factors to boost recombination to a very high scale. And that SNI1 was identified as another modifier. And with this, I want to finish. I just want to acknowledge, show my lab. This is maybe not very present picture because due to pandemic that is of course affecting the whole world. we couldn't meet to take pictures very frequently. Oh, okay. So I was showing mostly the data presented by some of the people here. I will not go maybe into details because this last project was not really shown, but it was also done in collaboration with Ian Henderson group from Cambridge University and with Monica Pradillo group from University of Madrid. And with this, I want to thank you for your attention, and I'm happy to take the question. Thank you very much.
Thank you, sir, for sharing the valuable information. Indeed, crop breeding is an area which is known since ages. Yes, yet it is an area with the constant scope of improvement. I thank you, Dr. Piotr, for sharing such valuable information on crossover control as the new breeding strategy. And now I would like to request our participants that if they have any query, please write in the chat box section. Dear participant, please post your queries in the chat box section.
Meanwhile, may I ask one question?
Yes, sir, please.
Thank you, Professor Piotr, to accept our invitation and to coming here. And because today is national holiday, National Independence Day of Poland. So I am also giving you wishes for that. Happy National Independence Day for the Poland, 11th November. So my query is that there is two strategy for crop improvement. One is using genetic engineering approach, and another is like this breeding approach. So what is your opinion on both of these strategies? This genetic engineering approach as well as this breeding approach using recombination frequency, increasing recombination frequencies.
Okay. Thank you very much for this question. I'm not really sure whether I heard everything. So I understood that you asked about what I think about GMO, genetic modification in plant breeding, and the traditional strategy for plant breeding. Yeah.
I am repeating my question. I'm audible. Professor Piotr. I am audible.
Sorry. Yeah, please repeat your question because there is a little bit problem with your microphone. I think. So.
Okay. So am I audible now? Am I audible?
Yes. Yeah.
Yeah. Okay. So the query is that there is two strategies. Like one is genetic engineering approach for crop improvement, and another is like this crop breeding approach using recombination frequency or like that. So what is your opinion on these both of the strategies for crop improvement, which is more nowadays, which should be focused more for real crop improvement in the field?
Okay. Yeah. So thank you for this.
And also I sorry for interruption. And also I said that Happy Independence Day, National Independence Day of the Poland, because this is 11th November. And this is National Independence Day of Poland.
Yeah. Thank you very much for today. Indeed, we've got the Independence Day in Poland. So actually, I'm staying at home, but I'm still happy that I could participate in this meeting. But coming back to your question, so I think that the food demand is really growing very fast, and actually we need to probably use both strategies to increase generation of new cultivars. So first of all, traditional breeding using crossover recombination, but before we didn't really control. it was like more random crosses of different plants. We couldn't do much to increase this recombination frequency. It's something that is still used and will be used, because still, even for using genetic engineering of plants, you need to cross later different cultivars, and that's what people are doing and will be doing for many years in the future as well. But there are two things with genetic engineering and, for instance, modification of the genome using CRISPR, genome editing, or transgenic plants. First of all, we don't know all the genes that are, for instance, responsible for resistant to drought, for resistant to different pests, resistant to high temperature or water deficit, that are currently very important problem for food production, and they are causing lots of limiting the production of food. And because we don't know the genes, we cannot modify them using genetic engineering, because to use GMO, we need to know which gene we should use. And in crops, it is impossible in many cases. We know, of course, some genes that are encoding resistant to some pathogens, but in many cases, this is not known. We could identify more or less the region which contains the gene, but we are not able in crops to identify the gene. And sometimes these regions are of, usually those regions are of mega base pair distances that contains thousands of genes or hundreds of genes. So we cannot use it in direct genetic engineering approaches that you mentioned. So, but of course, some of them we know. So I think what we should do in the future is to try to combine both strategies. Try to combine both genome editing and transgenic plants to, for instance, make sure that plants will be resistant to some insects that is commonly used at the moment, but also use this crossover-based cloning, the crossover-based recombination to combine different traits that are responsible for yield, for instance, production. Because one thing that I didn't mention is that we usually try to, it's relatively easy to manipulate with a resistant gene to some pathogens, but it's impossible to use this strategy to modify yield because this requires many genes at the same time. Thank you.
Thank you very much. Thank you, sir.
Thank you, sir, for addressing the query. And one of our students has requested you to kindly elaborate the advantages of crossover breeding over GMOs.
Yeah. Okay. So as I think that like I partially respond to this question by responding to this. So yeah, as I said, some genes can be used, can be modified by GMO, and I think that in these cases we should use this method. But in many cases we don't know the genes, and also many traits are encoded by huge numbers of genes that you cannot use in GMO approaches. In GMO approaches, we usually incorporate 1-2 genes at the same time, but we cannot incorporate hundreds of genes that would be required for changing yield efficiency, for instance, yield of the plant.
Okay, sir. One more panelist member has a query, and he has asked that what are your thoughts that this pandemic situation, will this situation has any effect in the field of plant breeding, as plant breeding is considered very crucial for food security? So, sir, what are your thoughts?
Yeah, well, I think that unfortunately, the pandemic situation that is affecting the whole world will negatively influence these things, because now we will have to spend more money to restore our economics in different countries. Also, both in rich countries and poor countries, we will need to speed up our work use resources to get back to the situation before pandemic. It's not easy, it requires a lot of money, and because of that, it can also affect the money that we can use for agricultural work and for food production. So I think that unfortunately, this will definitely not help with food security in the future. But of course, we need to do our best to solve these problems. Thank you.
Okay, sir. Thank you, sir. And one of our participants would like to know that whether they can produce double haploid brassica plant by using your approach.
Well, the approach is this approach will, of course, double haploid brassica are normally produced from pollen grains that are and with colchicine treatment we can duplicate the haploid genome to get haploid plants. I don't really think that this approach could be used directly, but of course, if you generate double haploid brassica plants that will be carrying interesting genes that are important for resistance and yield, then you can, of course, increase also recombination to faster generate a beneficial combination of algae, which would help to develop a new cultivar. So I think that in a long distance, yes, it should help.
Thank you so much, sir, for addressing all the queries of our participants so nicely. And thanks to both the keynote speaker of the first technical session for sharing the various valuable information with our participant. So now we will resume back after a short lunch break at 1:40 p.m. So now after 1:40 p.m. we will gather back here for the second technical session. So thank you all the participants, and thank you, sir, and thank you, Kalaivani ma'am, for accepting the invite and being here at this event.
I also thank you for invitation.
Thank you.
Thank you.
A very good afternoon to one and all. Myself, Dr. Shilpa Prasad, on behalf of ICCOB Organizing Committee, welcome all speakers and participants for post-lunch session. After a prolific session by Dr. Kalaivani Vivekananthan and Professor Paiter, let's continue with second technical session. And for that, I would like to introduce our speaker. It's my great honor and pleasure to introduce our esteemed speaker, Professor Malik Yusuf, Associate Professor and Head of the Galilee Digital Health Research Center of Zephyr Academic College, Department of Information System at Zephyr, Israel. In the last 20 years after doctorate, Professor Malik has published more than 80 research articles along with patents in his name. Professor Malik has done postdoctoral studies from University of Pennsylvania Center for Bioinformatics, USA, and Wistar Institute, Center for Systems and Computational Biology, USA. Professor Malik has earned several academic ranks in institutes of higher education in Israel and also contributed to various international forums and societies outside the institution membership. Professor Malik Yusuf is also member of editorial boards and contributed as a reviewer in several journals of repute. Till now, Professor Malik has been constantly bestowing in academic activities and active participation in conferences. Professor Malik has also acquired numerous research grants as principal investigator. Today, Professor Malik will be sharing his talk about application of biological domain knowledge based feature selection on gene expression data. I request all to please join me in welcoming Professor Malik Yusuf. Please, sir. Please, sir, you can continue. Kindly please unmute, sir. Yes.
So this is my turn. Hello.
Yes, sir. Yes, sir, you are audible.
Okay, do you hear me?
Yes, sir.
So who's supposed to talk, me? Okay.
Yes, sir.
Okay.
You're audible.
Okay, thank you very much. So thanks for having me and invite me to this great opportunity to talk about our different research, which is mainly about integrative approach. We actually developing different algorithms in the field or topics of bioinformatics. Actually, we are presenting novel and a new approach of feature selection, which is based in grouping and scoring components. So, so actually for this purpose, I would refer to you to some review that we wrote in this topic. The review, if you are interested to read more about the integrative approach, is called Application of Biological Domain Knowledge-Based Feature Selection on Gene Expression Data. This work was done by me, Omar, Abishka, and Borjo from Abdullah Gül University. We actually survey different approach for feature selection. And also we talk about our different approaches in this manner. It's very important to indicate that the integrative approach has become very popular now and it's gaining attention. Why that? Because let's assume that we have a gene expression data, which is actually a tabular data. It's a matrix of numbers. So if a machine learning guy see this kind of matrix, he will immediately think about feature selection procedure in order to obtain the most significant genes or the most significant features. I can use both terms. So in this matter, we have a matrix that we would like to find the genes that actually somehow form a biomarker in order to detect and classify the cancer from healthy, or any two topics or two groups or classes that we have in our data. So what is the merit of our approaches? So most of the existing feature selection approaches actually have been borrowed from the field of computer science, because that I said, when the machine learning guy or computer science guy see this kind of matrix, then he suggests, oh, you should somehow apply some machine learning algorithm or feature selection with machine learning. But actually, most of these approaches did not consider the biological domain knowledge, because actually we have a biological data. Somehow we need to think about how we can integrate the biological information into the process of the machine learning and feature selection to get maybe to improve the performance and also the biological interpretation, and also suggest a new outcome that is not suggested by the traditional approach. So actually the traditional approach of this kind of machine learning feature selection applied to this kind of data is to perform some data pre-processing for a given data set. Let me try to make some annotation. Okay. So if you look to here, actually you see there is the pre-processing step that we try to do some normalization, quantile, PCA, and so on and so on. And the other thing that after that, we can apply different machine learning approach like feature selection, like also classification and class discovery by clustering, unsupervised algorithm, doing some cross-validation. There is a lot of approaches that are existing in the field in order to perform this kind of feature selection and machine learning and build the algorithm in order to predict. And then what these approaches are doing, in the end, they actually suggest a significant list of genes, which is actually a list that we take after that to make some biological interpretation. So if you look to the process, they are separated. The machine learning algorithm and the arrangement process, where we actually take the list of significant genes and then we go map it to some public database like mutare-base, cake disease, SNPs, methylation, and then actually we try to come up with biological interpretation. And again, if you look to this two processes are somehow not connected. So the question how we can embed this kind of knowledge into the machine learning. So this is actually our aim, is to embed this biological knowledge into our classification task. Actually, this is the purpose of all our research. And I can say also we dealing with other kind of data in the era of multi-omics, and also we suggesting the different approaches now, even we're writing some grant for this purpose. Okay. So biological integrative approach take advantage of multiple published genes, expression data set. The integrative analysis of gene expression data has become an effective tool by aggregating multiple data set and increasing the statistical power and identifying a small subset of genes to effectively predict the type of this disease. Again, this was published by Yang, Z, and all these guys, multi-view based integrative analysis, and they also emphasize the importance of using integrative approach in order to increase the statistical power. Okay, so the idea is we can summarize the idea in this figure or this workflow. Just to try to explain it, because actually most of our tools are relying in this figure. So in our approach in general, I'm now talking about biological knowledge. In the end, we need to specify which kind of biological knowledge. It's a microRNA target, a methylation, KIG pathway, and all this stuff. So in this things, actually, we have, we assume that we have this kind of knowledge, the gene expression data that we saw before. And then we need to integrate biological knowledge that actually represented as a database. So we assume in our generic approach that there is two kind of data, the gene expression data that actually everybody is using, and we have the biological data, which is actually the domain biological data represented as a database, like mutare-base and so on, so on, if anybody knows this kind of database. Actually, our approach is mainly consists from three components. The group, we call it G, the scoring, and the machine learning, the M. And if you look here, you have the G, the S, and the M. And the end, we have output, which is actually the output of all this process. I would like to emphasize that there is something new in the output that does not exist in the traditional approach. The output layer actually gave a new way of performance table, and I will explain it later on how this performance table is look like. And additionally, the traditional approach suggests significant genes, but in our approach, we suggest significant groups. This is something new in our approaches, that actually we suggest not the significant genes, we suggest the significant groups, because the group actually is associated with a specific biological knowledge. It could be, for example, this group of genes associated with some specific disease. So actually we look to the features as a groups, not as individual. And the groups make sense. Then the biology take this output and try to make sense of this kind of output. So in our generic approach, as I said, we have the BioD database and the gene expression. And in order to do some validation, we split the data into training and testing. And from the training, we use the biological knowledge in order to create the groups of the genes according to the BioD. And then actually, this is the groups. We perform some scoring matrix in order to score the importance of the groups in terms of classification. And then we create a model. The model will be tested by using the test group. In the end, we decide to use random forest, but anybody can also suggest another approaches for this classification. So actually, this is the generic approach that we suggested. So in order to explain what is the group actually, for example, let's assume that we have knowledge about microRNA target. So this is, for example, extract from MIR-TAR base. And this MIR-TAR base is a biological database that has information about microRNA and the genes. And again, in our data, we have information just about the genes, the gene expression data. We don't have any information about actually the, we have just the genes and the expression of the genes. This is actually the gene's name. So in our approach, the knowledge that we have is the genes expression. So we use the biological knowledge, which is, for example, microRNA. So this specific microRNA target this set of genes. So actually, this is kind of knowledge. We use it in order to perform the groups. So actually, in this way, we use the microRNA target, that database, in order to create the groups. So now, actually, this is just an example. We have how many groups? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 groups. So each group actually has a name, which is the microRNA name that we extracted from the MIR-TAR base, and a list of the genes that this microRNA target these genes in terms of biology. This microRNA actually target these genes in terms of biology, and so on and so on. So the question, when we have the genes, so how we can actually score it? For example, the groups actually we now extracted, we created. So the second question, how we apply the scoring component. Now the groups are ready in this way. So in order to perform the scoring, our approach is suggested to do the following. We actually, from each group, we extract from the original data its subdata. This subdata just contains the genes that associated with this group. For example, HSA, LET7, A5P. So now we have only the column, which is the genes, and the class label, the original one, that we extracted from the original data. So now this subdata represent this group. So actually, if we have subdata with label, it's easy to perform machine learning. So now we extract for each group its own subdata that contains just the genes belong to this group, if you look to the examples. And then we have different kind of subdata that we can apply some scoring metrics. In our approach, we suggest to use machine learning in order to do that. So in our approach, actually, we partition the data, again, the subdata, into training and testing. And then we run decision tree or random forest a couple of times, R times, in order to give a score. So actually, in the end, we will have some knowledge about these genes, set of genes, in terms of classification of the original data. So in the end, actually, we'll have some scores for this kind of groups. So in simple word, I explain the grouping, the scoring, and then we need to build a random forest in order to test it. Okay. So in our research, we categorize the integrative of biological knowledge grouping in 3 categories. The one actually is group genes based on biological knowledge. For example, we know that genes that have similar function are correlated. So according to this biological knowledge, we use, for example, clustering for grouping. For example, SVMRC, it's a tool that we published. SVMRCR, it's advanced. For this tool also we publish it. SVMRNE, also another tool that we publish it, and is available over our GitHub. Another kind of a group is using some database that contains this kind of biological knowledge, like KIG pathway, gene ontology, MIR-TAR base, and disease. This kind of tools, actually, we publish MATI, Cognet, gene ontology, and 1, 2, 3, 4, 2 is under development. The other one is group genes based in 2 or more omics dataset. So actually for 2 omics, we publish this one, and this one is under review. And now also we're developing a new approach that we call it KMint. KMint, it's a new approach that we are developing. It's a project with UNC, Professor Meron from UNC, University of North Carolina, and Dr. Borjo from Abdullah Gul University. Our student, Mirai, PhD student, she's responsible about this new approach that we are developing just recently. Okay, so actually we divided into 3 parts. And again, for example, let's look to the different approaches just to survey it. And actually I just make a reference for each one if anybody interested to have more information. So the SVMRCE, the first actually study we suggested in this topic called SVMRCE, Support Vector Machine Recursive Cluster Elimination, is published in BMC Bioinformatics in 2007. Actually, this is the first study that was to suggest the approach of dealing with genes as a groups. So in terms of groups, actually we use the clustering like k-means in order to create the groups. And then we have different kind of groups. And we use the R or score in order to scoring according to the procedure that I just described before. But this is actually a unique algorithm that we perform also recursive cluster elimination. So actually when we group the clusters, we have some score for each importance of the significance of each cluster, and then we eliminate the least significant cluster. We go back again, we perform the cluster until we end up with, for example, 2 clusters or so on. This is actually the details of the algorithm in this workflow. So in terms of output, we are not just plotting the output of the performance. We give a new insight of performance, a new kind of performance. We give the number of clusters, the number of genes in these clusters in average, we perform that 100 times, then the accuracy and so on, so on, so on, all this measurement. And then also cumulatively, we give when we have 2 clusters, when actually we have 159 genes, this is for a specific data set, and this is the performance. And we do that process for actually 90 clusters. So why 90 clusters? Because we start with 100 clusters, and each time we reduce 10% from these clusters. Because that actually, each time we reduce it 10% until we reach 1 cluster. So this is actually the performance table that we represent. Additionally, we also keep track about the genes and significant genes that they are show up to be important. Later on, I will show another approach that we score the genes. Okay. Okay, actually we did some comparison with other approach showing that SVMRC work very well and even getting very high performance. And also we show the difference in terms of the significant genes between SVMRCE and SVMRFE, and suggesting that SVMRCE giving a clusters of genes rather than just a list of significant genes in this way. So during this study, actually, we suggest that gene interaction networks or other biologically relevant metrics that group genes based on functional parameter might also be useful. So actually, this is, we wrote it in 2009 and 2019. Actually, we suggest the MATI algorithm. But again, after that, also we published another study in BMC Bioinformatics, where actually use GXNA. It's a tool that actually use the gene expression in order to create some networks, gene networks. So each gene networks actually serve as a group. And then we perform similar to SVMRCE. And this is actually also, we compare it with other approaches like SVMRNE, SVMRC, the one that we published before, SVMRFE. SVMRFE is a support vector machine recursive feature elimination. And we show that our approach also working well. And we give some information. Okay, the second part that we have 3 categories. The second part actually is just wait. Okay. Okay. So the second part was actually to use biological knowledge in order to perform the grouping. And this study actually was published in Bioinformatics. The idea is to use MIRTAR base, which is actually host the information about microRNA and targets. And we can it as a table in this form. And if you look to this workflow, actually we have as input a gene expression, and we'd like to add knowledge to this process by using MIRTAR base. This was the first study that we combined biological database knowledge with gene expression, and actually this is the kind of a group that we have. This study actually, as again, was published in Bioinformatics. The approach called MATI, discovering target interaction. So what is unique in this approach? As before, the new thing in this approach, actually, this is the performance table. And again, this is the tab group, which has 3.8 in average genes. This is accumulatively the tab 2, 7.5, tab 3, and so on until we reach 10 groups. And actually, this is the significant genes that every tool suggesting. The unique thing in this approach, actually, we produce a new kind of output. We rank the microRNA. Even the microRNA is not part of the gene expression data. According to our approach, we are able to group, actually to rank or score the groups that we created from our approach. So HSA-MIR 145-5P, they seem to be very important in this study. So actually we add additional biological information that was not exist before. So if you just look to the input, the additional input is somehow gene expression, and we adding a new biological knowledge to the output that was not exist. This is very, very, very novel thing actually we suggest in our approach. So now the biology look to see why this microRNA is important in this term of this data. Additionally to this list significant genes. So keep in mind that we add biological knowledge that was not exist before. Okay, this is actually, we will use it later on. Okay, I will not talk about the more details because actually the paper is available. I would like just to show you the importance of these tools. And again, another tool called classification of gene expression database or ranked active subnetwork-oriented cake pathway enrichment analysis, the tool called Cognet. Actually, we give the name Cognet. So Cognet actually use another tool which called PathFindR. PathFindR actually getting as input a gene with p-value, gene list with p-value. So actually we use the gene expression in order to give this information to PathFindR. The output of PathFindR is a somehow significantly enriched pathways. So actually create some N data set for each pathway, which is actually, this is the groups part. So in our approach, we use PathFindR to perform the grouping. And then we proceed with our scoring and the M score, M component. And if you look again, this is actually the traditional approach that we suggest some performance table, significant genes, but additionally, we add the importance of each KIG pathway, which is not part of the gene expression data. This is something very important that we add a biological knowledge, but just by integrating this biological knowledge to our machine learning algorithm. So now we have also important of each KIG pathway in our list. Okay. How many time I still have? How many time I still have? Sorry? I have 10 minutes.
Yes, sir.
10 minutes?
Yes, sir. 10 minutes are there.
Okay. So actually, again, we compare the Cognet tool with other tool, for example, with SVM-RCE, with MATI, and Cognet Random Forest. We saw the average AUC, the number of genes, and you see Cognet sometimes actually outperform, for example, MATI, but SVM-RCE outperform both of them. But again, this is not the idea of Cognet to compete in terms of performance. The idea is to suggest another way of looking to this kind of data set. And also we perform some validation and so on and so on. So actually, this is the Cognet. The third category, if you remember that actually we talk about 3 categories in our talk, 1, 2, 3. So I present one of this kind, MATI, Cognet, SVM-RCE, SVM-RNA. And the last kind of tools of combining is using multi-omics. So in this approach, actually, we have just 2 omics data set. Gohan from Abdullah Gul University with Dr. We conducted this study, and the study just published in PeerJ. And also we have a collaboration from Jefferson, Philadelphia University, Thomas Jefferson University, Remy Krenschka and Christian. They actually contribute to the biological interpretation of this tool. Okay, so the idea here is how we can use this to multi-omics. So the given data is actually microRNA, which is actually gene expression of microRNA, and gene expression, regular gene expression. And this is actually the gene expression of the microRNA. To make it simple, gene expression. So how we can, actually what we have done in this study, first of all, we found the expressed microRNA and the expressed microRNA, and just be noticed that we apply this in the training part. So then actually we know that microRNA actually target genes. If they target genes, they should have some correlation, or they highly correlated or low correlation, right? So or they highly correlated, I mean in the term of reaching +1, or they also highly correlated reaching the -1, different conditions. So in this way, actually for each microRNA, we find all the correlation with the messenger RNA. And this way, actually, we create a Pearson correlation coefficient for both data set. And then actually we detect microRNA and microRNA target groups. In this way, actually, we say if we set some threshold, and according to this threshold, we detect which microRNA actually has its own target sets of genes. And then we proceed with the rank and so on and so on. So actually, this was the approach. And again, this is the workflow that explain each step in our MIR-CorrectNet. And again, we create some subdata that each subdata is moved to the rank or the score component. And then we have a scoring matrix for each microRNA. And this is the target genes. Okay. And as before, if you look to the output, again, we have the output of the top significant groups and actually the significant genes. Additionally, we give information about the microRNA and the list of the genes that they are associated with this microRNA. And we give some p-value according to robust rank aggregation package. In this time, actually, we integrate this package that actually know how to integrate different significant list, ranked list in order to integrate it in one list. Because we actually repeat our algorithm 100 times. So each time we will have different kind of scored list. Okay. So in the end, also we compare our approach with MATI, MIR-CorrectNet, ASVM-RSCE, ASVM-RFE, and we apply this approach in data that we get from TCGA website, and we report about the performance about different disease, breast cancer, lung cancer, and so on, so on, so on. This is actually the tool that we coded in K9, also available our GitHub. So as I said, these two researchers were involved and they wrote the biological interpretation of the results. And interestingly, we found that MIR-21-5P was actually in the top of the list when we did the whole analysis, and it somehow fit with the prior knowledge about this microRNA. Actually, our tools is somehow validated even knowledge that exists in this kind of data set. And we did some validation. Okay, so the other approach that also was just published in DIXA conference, we continue this project. The project with Borjo and Ahmed is to integrate the gene ontology as we have done before. And again, similarly, now we have gene ontology list of genes. And we also did the same thing in terms of ranking, scoring, and so on. And another work with Abhishka Kumar and his student Yushman. The tool is ready. We just need to finish the manuscript. The idea is how we can combine, integrate disease association with the gene expression data. And also we have very interesting results. And the interesting thing that sometimes if we, for example, study data that actually talking about a couple of the disease or one disease, then actually we see the genes that are associated with other disease are related to this KIRG disease. So it will be interesting to see the connection between different genes that are related to different disease to a specific data set that actually is subject for a specific disease. All these tools are actually available in GitHub. You can download it, can use it. We will be happy to also talk about collaboration. And all of these tools mostly written in K9. Thank you for listening. And I would like to thank all of these people that were somehow involved in this project, Jens, Larry, Luai, Luis, Borjo, Sezerman. I just talk about the BI. And thank you very much, and ready for taking question.
Thank you so much, sir, for such a wonderful session. Now the session is open for questions and queries. Participants can post their queries in chat box.
Meanwhile, may I ask my query?
Yes, sir, please.
Yeah, it's a very nice and very informative session, and a lot of tools we were kept to know in which we can work a lot. One tool I found very much interesting and related to my work, that is MIR-CorrectNet. And so I want to know that, for example, on the scale of 1 to 10, what is the accuracy level of the prediction between the role of microRNA and their target gene expression? Means accuracy, what is the accuracy out of 1 to 10 scale of this tool?
What's the accuracy?
Accuracy of the prediction.
It's very high. It's very high. Okay. Actually, it depends. First of all, you know, it depends on the data that you are using.
Okay.
For example, MIR-CorrectNet performance, this is the area under curve, and you see the performance is very high. So, but again, this depends on the data that you extract. For this kind of data, we extract a patient, and you know, in TCGA, there is just patient. but there is patient with very small lung or very small modulo. So actually, according to the specification of TCGA, we use this kind of patient to be control, and the other one we use it as a patient. But we now trying to run this kind of tools in other kind of TCGA data, for example, from different kind of breast cancer subtypes, and to see how our tools is working well. But for this data that we have, the performance is very high. And again, this is not just the performance, the output is the most important, that you now see additional information that was not exist before. For example, the significant genes of microRNA that is added to this biological knowledge.
Yeah. Thank you. Thank you for this clarification.
Okay, thank you, sir. We have one more query from our participant. He has asked that according to what you said, groups are more important than individual genes. So can we test the effect of changing one gene and the effect of pathway regulation by using this tool?
Yeah, this is a very important question. Thank you very much. Yes, it's very interesting to examine these tools by, for example, changing genes and find maybe super genes, and maybe taking one gene out and see the model. Yeah, it's very, but you need to update the tool, but it's very nice idea for a research to see the importance of genes in terms of groups. So taking these genes out of the group and then run the tool to see if this specific gene is important. And actually we have different idea how to score the groups, not individually. So for example, you can take the mean of these genes in order to represent the super genes. Yes, I think it's very important to also shave the genes inside the cluster or the group to see the importance of the group and minimize it to the smallest number of genes that can contribute to your classification. This is a very nice idea. We have it in our list, but we don't have time to implement it. This is a very nice question.
Okay, so if any more participant is having any question, then please post it. Okay, I think there is no more query. Thank you so much, Professor Malik Yusuf, for sharing this piece of information with us in this platform. And now I would request Dr. Rahul to please continue with the next technical session.
Thank you, Silpa ma'am. Am I audible, ma'am?
Yes, sir, you are audible. Perfect.
A very good afternoon to all academicians, scientists, scholars, students, and dear Amitians present here. On the behalf of ICCOB 2021 Organizing Committee, myself, Dr. Rahul Kumar, would like to thank Professor Malik Yusuf for the great talk. You enlightened us with your vast knowledge and experience in computational biology. Now, I would like to welcome Dr. Vinay Kumar sir here. Just a brief introduction about him. He is serving as an associate professor, Department of Biotechnology, Progressive Education Societies, Modern College of Arts, Science and Commerce, Pune, and also serving as a visiting faculty in Department of Environmental Science, Savitribai Phule University, Pune. He received many awards and grants like Young Scientist ACRB DST in 2012, member of DBT for Star College Proposals 2018 to 21 till now, member for DBT Builder Program, DST Feast programs, etc. Also, he is active in international recognition like member of Asian Federation of Biotechnology South Korea, member of Asian Council of Science Editors UAE, and senior member of Chemical and Biological Environmental Engineering Society, Hong Kong. He has guided 4 PhD students and 2 students having their PhD ongoing under him. And more than 50 PG students for their research programs like 6-month dissertation, and more than 30 graduate students for their short-term research projects. So I would like to welcome again Dr. Binay sir. Please share your PPT.
Thank you, Dr. I hope I am audible to everyone.
Yes, sir.
There was some technical problem in the morning, so I was thrown out of the meeting. I apologize for that. On the onset, I would like to express my sincerest thanks to all the organizers, all the authorities of Amity University. And since we are now collaborators also, though we have to start our activities together, but I think this is a nice beginning to come along with you people and to interact with you. And I hope that after a wonderful session by Professor Malik, I'll maintain the tempo. Thank you once again. Thank you so much for inviting me and having me for this wonderful opportunity. And I'm sure my talk will help the newcomers, young faculty members, as well as students to start working if they are not, and to strengthen their work if they are already working on microRNAs. May I share my screen now?
Yes, sir.
Is it coming?
Yes, sir.
Unfortunately, some part is going, behind the images. Can we do something? If you can help me out. Can you see the full presentation plus video?
Yes, we see it. I think it's okay.
It's okay.
No, unfortunately, in my screen, I can see the screen, but there are some, the panel of videos also on one side. So I may lose some content during the,
I
talk.
think we can see your complete slide here.
Actually, Binay sir.
Yes.
In the view option, you just Binay sir.
Yes.
Am I audible?
Yes, you are audible, sir.
Sir, am I audible?
Yes, sir, you are audible. Can you hear
in the view
me?
option, you just go into fit to window option. Just click in the fit to window. Once you will share the screen, it will come at a view option. There you will see zoom ratio, that is fit to window, and you just click in the fit to window portion.
What's the scenario now?
Now it is okay. We are seeing the exact way.
And video
No
also.
Yes, sir.
Okay, perfect, sir. Thank you so much. So after this brief, trouble, let us start with the talk. So my talk would be themed on microRNA biotechnology for climate smart crops. So what I would be talking about in next 30-40 minutes would be how the changing climates and challenging environments are impacting our crop production and quality of produce, and it's a major challenge for food security programs, including sustainable development goals. Then how microRNAs are emerging regulators of gene expression, particularly during plant growth, development, and more specifically on the stress responses, followed by different approaches. If someone wants to work on microRNAs, what are the ways and approaches for identification as well as validation of microRNAs and their targets? Then finally, what are the approaches one can study the microRNAs which are implicated in environmental stress responses and adaptation of the plants. And now we can explore these microRNAs if somebody wants to develop some climate smart crops. So as we all know that the plants are sessile and organisms, means they cannot run away. Unlike humans, they have to stand and deliver and face the conditions, whatever they get or whatever is thrown to them. That may be a changing climate. Even under normal condition, if you look at a plant, it faces so many difficult situations throughout the day in 24 hours. Sometimes it's high sun, sometimes it's dark, light, and so many things they have to face on. But in recent years, particularly driven by the anthropogenic activities, the plants are facing these problems plenty. And because of these conditions, the crop plants often get exposed to very harsh and fluctuating environments. And these days, you must be seeing that initially people were studying only the single stress, but now there is an era where many research groups are engaged themselves, they are engaging in studying dual stresses, multifactorial stresses, or the sequential stresses in an attempt to mimic the natural conditions which plants often face in their life cycles. So the climate change and the environmental stresses, they are putting lots and lots of pressure on plants. And all these fluctuations and changes are driving irregular water supplies that may lead sometime to the drought conditions. On the other hand, that also may lead to the waterlogging condition. Then hypersoil salinity is one of the biggest problems which we are facing globally, and plant growth is particularly hampered by the high salt contents in the soils. Then there are temperatures which are always extreme and variable in and around a different kind of field environments, then UV radiation and heavy metal stresses. Now, beside that, there are many other xenobiotics also that are putting lots of pressure on plants, including the nanomaterials which we heavily are releasing into the environment. Okay, so how plants cope up with these conditions? एक कहावत है हिंदी में कि प्यार और जंग में सब जायज है. Everything is fair in love and war, and that's how plants also respond to the different conditions. So if they want to attract someone, they produce several different kind of metabolites, aromatic compounds, wonderful flowers, tasty fruits, and so on. But if they have to cope up with harsh environmental conditions, they also produce several different kind of metabolites and osmolytes, which they particularly help them to survive under those difficult conditions. And all this is managed very delicately, very finely at the genetic and genomic level, and of course these days as we are studying at epigenetic level also. So in that, what plants do, they try to manage, they try to respond to these environmental conditions through genetic reprogramming. Means what? That they, very fine-tune the expression levels of several key genes. And this regulation happens particularly at transcriptional, post-transcriptional, or translational levels. And microRNAs in recent years have emerged as critical post-transcriptional gene expression regulators that may be required by the plants to attenuate the plant growth and development stages and the conditions and the goals under stressful conditions, beside from the recovery also. Okay. You must have heard about different type of terms when we talk about microRNA. Sometimes we hear about RNA interference, sometimes we talk about RNA silencing or gene silencing. And many a time these terms are intermittently used here and there. So RNA silencing is or gene silencing is a way where small known coding. Now I'm emphasizing on known coding. These are very small known coding, particularly for the students. the RNAs which do not encode any protein. Initially, this kind of DNA was known as junk DNA, but no more. Now we know that all different kind of genomic stability, particular to the higher organism, is given by such non-coding regulators, and these are very, very important. And according to their origin, processing mode, and effector protein association, these small RNAs can be divided into different categories. Today we'll talk more about microRNAs. And microRNAs are predominantly, gene suppressors which suppress the target genes post-transcriptionally. These kind of studies, whatever the data which I would be using more or less would be from our own published papers in recent years. We started with plant microRNA work in 2016, and we published this in Frontiers in Plant Science in that year. Okay. So for the young ones, those who are new to this world, I would like to tell them about microRNAs. So they are very, very small, tiny molecules, roughly from 19 to 24 nucleotides, and they are found abundantly in plants, animals, and of course in some viruses also, and bacteria. In terms of microbes, we usually call them small RNA. So they function in RNA silencing and post-transcriptional regulation of gene expression. And through this base pairing with mRNA or the transcripts. Now I would like to tell one thing here, that miRNA always target mRNA, so they are like Jani Dushman of each other. So on a lighter note, so as a result, these mRNA molecules are silenced. Means what? The genes, either they will not produce any transcript or protein, or the process won't be complete. So there may be a cleavage of mRNA strand, there may be a destabilization of mRNA, and then less efficient translation. Again, that will hamper the production of proteins by the ribosomes. We published this data in 2020 in the journal Planta. Okay, so just a very, very brief overview how these miRNA get synthesized in plants or their biogenesis, and what are the general mechanism of miRNA mediated regulation of stress responsive gene. So here in this picture, we can see that this is an miRNA gene, and through different processes, it will form first primary miRNA, then pre-miRNA, and finally the miRNA-mRNA duplex will be formed. And this is the mature miRNA, and it will make a system here that is known as RISC. As the name suggests, it's very risky for proteins, and it does not allow the proteins to be formed. This is known as RNA-induced silencing complex. And through different type of processes like translational inhibition or mRNA cleavage, they may hamper the transcription. Okay. I would also like to tell here that miRNA, though people historically feel that they are negative regulators, but not necessarily, they may also be positive regulators of plant growth and development, and particularly under stress tolerance. So it was all started in 1990s when Rosalind Lee, they reported first in the nematode, but it went totally unnoticed for next 10 years, and it was all in the earlier 2000s that people started realizing the potential of microRNAs. And at the same time, it was also started in plants. So if somebody asks that how the miRNA are important for the plants, where they are involved, so literally speaking, they are involved everywhere in all the life processes of plants, plus their responses to different environmental conditions. One interesting fact about microRNA is that one miRNA can have multiple target genes. Means what? One single miRNA can target many genes, and the vice versa also. Both are, possible. So if somebody wants to work on miRNA, because this general talk anyone can make, we can also find plenty of information on Google and in different review articles. But particularly if some students, young faculty members or young researchers, they are planning to start work on miRNA. Though I'm talking about plant, this is more or less same about all the living system. barring some exceptions, some changes that you have to take care of when you are changing the system, but more or less the workflow would be same. So it always start, or this is, I would say, a standard method. We have published this kind of methods standardized in different articles. Like in 2020 we published in Acta Physiology of Plantarum, and if somebody is really interested in working on microRNAs, he or she can directly contact me, and definitely we would help you. So it starts with isolation of RNA, and then you can prepare the RNA or small RNA libraries. Then small RNA sequencing can be taken place. These days, Illumina HiSeq is well in fashion, I would say, and heavily used for small RNA sequencing. And through that, you'll get millions and millions of reads, which we call as raw sequences, and all the unnecessary things will be removed. There are different tools available, and many of these tools are freely available or open source, which can be used to remove these adapters and other unnecessary things which you get through the sequences. After getting that, there is something like Bible in miRNA work, which is the MIR base. This is the base of all the miRNAs which are reported so far. It is hosted by Sanger's Institute. And once you get this information through the small RNA sequencing, you can use the MIR base very effectively for searching and hits in the miRNA databases. Besides, you can also go for the reference genome. Reference genomes are very important, and that's how you select even the plants. As far as my lab is concerned, we are currently working on microRNAs from rice, microRNAs from soybean, and now we are also starting on the microRNAs from the cowpeas. And particularly these microRNAs which are responsive to different environmental stresses. Initially, we were working on single stresses, but now we are working on the combined stresses also, because we want to target this climate change and environmental challenges, and how we can develop climate smart crops. Climate smart crops are the crops which can cope up or survive under harsh environments and changing climates, as we have so many predictions and projections that the environmental changes are going to be more and more into place. And we have started experiencing, look, the climate has changed drastically. In the drought-like areas, including Rajasthan, there are heavy rains, whereas on some areas there are drought-like conditions. The precipitation cycles are also disturbed in areas like Maharashtra, where coastal lands are available for rice fields. We are finding increasing salinity plus temperature, a big menace for the farmers, and they are really challenged by these conditions. So I was talking about these. Then you have to predict the miRNA where you have got a new microRNA or you have got a known or conserved microRNA. After that, there are online tools available that will help you in identifying the targets. Then also the validation of targets can be done. And after that, you can introduce again the wet lab experiments. And through that, you can go ahead with the expression analysis of your microRNAs depending on your work nature. I hope things are clear so far, and it's not getting very sleepy.
Yes, sir. It is interesting, sir.
Okay, thank you so much. Okay, now I would like to share one of my own work. This work is on the rice varieties, particularly two rice varieties which are very popular in Maharashtra. One is salinity tolerant and another is salinity sensitive. So what was the base of doing this research? This research was funded by DST, and what we tried to see was that how sodium toxicity induced changes at post-transcriptional levels in rice, particularly at the reproductive phases. Because unfortunately, a large chunk of studies, you would find that people are engaged in studying different environmental stress responses in plants at vegetative levels. There are only a very few studies, and on these aspects there was no study. This was first of its kind of study, and that's why we could get the funds also. So we selected this mill stage, which was roughly around 105 days from the germination. And to study the sodium toxicity in rice, we introduced two different type of, stressors. One was sodium chloride, that the common salt. Beside, we also introduced sodium gluconate. That's a specific molecule which only allows the entry of sodium ions. So 100 millimolar concentration was that will give you roughly around 10 decisiemens per meter electrical conductivity of soil. So as I told you that we started with isolation and purification of total RNA, then we prepared small RNA libraries, and here which type of methods of next generation sequencing you are using is very important, because resolution is very, very important. You should get the desired results single end, and the resolution should be very good enough so that you are supposed to get 30 million reads per sample. Now, this is specifically important for students. If you are working with RNA and you are isolating RNA, you have to maintain the quality of RNA. And how you can check the quality? You must have heard about spectrophotometric analysis, where you are checking it using 260 to 80, but I would also suggest you to check with 260 to 30 ratio. And you can also check the quality of your RNA on the denaturing agarose gel. Okay, since this is a very costly affair, you have to put lots and lots of money in this kind of research. So initially we thought of, we have to find out whether the desired stress was introduced or induced to the plants or not. So we took 5 known marker genes which are known for, their role in stresses, particularly in plants, including rice. And when, we, conducted this experiment, we harvested the samples after introducing the stresses or after subjecting the plants to the stresses. And then we checked the real-time, PCR-based relative expression analysis of all these 5 marker genes. And since there was the differential expression of these marker genes, it was confirmed that the plants are experiencing the stress conditions. So first objective was, achieved. Okay, then we conducted, or we taken out the small RNA library preparation. There are known methods which are available with us. We also can share if somebody is interested. So immature grains were harvested, total RNA was, isolated. Here, if somebody is trying to work on small RNA or miRNA, one more parameter is very important, and that is RNA integrity. So you have to have a very high RNA integrity if you want to have the good quality of results. So your RNA, or sorry, RIN value, which we call it, RNA integrity, it should always be more than 5 or 6. Then only you can, or you should go ahead with the sequencing. There are some other ways also to check the quality. And through that, you get to know whether the RNA and the small RNA libraries are of good quality and whether you can go ahead with the sequencing experiment. So after sequencing experiment, we got, as I told you, that millions and millions of reads from both these varieties under all the conditions. So out of that, we identified 140 different microRNAs by comparing them to the known databases and by referring to the reference rice genome. Luckily, rice genome is available, that too with a very good resolution, so things are easy, and these studies were published in Science of the Total Environment. Again, these are the steps if somebody is interested. What are the analytical steps and what are the database and tool available with us, which can be used for such kind of study, which since we have limited time here, maybe some other time we can have this in a detailed discussion about that. Okay, so these were the 140 microRNAs that were differentially expressed, and that's how we identified how many and which of them got overexpressed and which of them got downregulated. So out of that, putting some more parameters and filters, we identified some 10 to 15 microRNAs, or 13 to be very specific, and we identified which are upregulated, means the expression level of which microRNAs has got up, whereas the expression level got down of which kind of microRNA. So here one can very easily correlate these results, that this salt tolerant variety is showing more upregulation, whereas this salt sensitive variety is showing downregulation of many microRNAs. That was a fair enough conclusion. And by putting rank aggregate analysis, as Professor Malik was also targeting, we also use some R-based tools which tells you the rank of your identified microRNAs. So we got this top ranked or top responsive microRNA, that is MIR1861E. After that, the main target or the next objective for us was to identify the targets of differentially expressed microRNAs. So though we selected only one, but we conducted studies for all these microRNAs, all 13 microRNAs, and then procurement of accession numbers of identified targets and identification of gene ontology annotation. So you have to characterize all these microRNAs, what the genes they are targeting, which class they belong to, and these are the results of gene ontology analysis. First, let's see the salt tolerant variety. So here you can see the cellular components, then the molecular function, as well as the biological process where they are involved into. So through that, you'll get all the necessary information as far as gene ontology is concerned. This was followed the same in the salinity sensitive variety also. Okay, so ultimately this is the summary of the top responsive microRNAs, as I was telling you. So what kind of cellular component it take part in, what are the molecular function, where it can have some roles, and what are the biological processes, and how many genes it may target, or it may be targeted by. Okay, so this analysis told us that the targets of these top responsive genes are multifold. As I was telling you in the initial part of my talk, that a single microRNA can have multiple roles. That's why the microRNAs are becoming so important. Friends, in initial years of biotechnological research, people were only talking about the gene which are encoding proteins. And that's why the transgenesis or the transgenics were produced using single or maybe double or maybe 2-3 genes, which are known as gene cassettes. But after that, we realized that particularly this regulation is very important, and the regulators, that too microRNAs, have very important roles. And that's why many research groups across the globe, they are trying to identify different microRNAs which can be explored for different plant species, because these microRNAs are highly conserved, and that's why the chances of their success in different plants is very high. So here you can see that it has multiple targets that are correlated with the stresses, the phytohormones, the transporters of different molecules, as well as the transcription factors. So it covers a very good picture. Okay, so these targets were validated further. There are ways to validate the targets, and we identified that 2 very known one enzyme and another protein which are known for their stress roles were identified and validated. After that, this validation was done using wet lab experiments, particularly by RLM-arrays PCR reaction. And after this, we got the purified PCR products and we cloned them, and then the sequencing was carried out. So we got the validation perfectly fine with the RLM-arrays technique, and these are the results. How much more time I have? 5-10 minutes?
Yes, sir, 10 minutes.
Okay, so we can conclude from these results that the gene annotation for all the target genes of the microRNAs which were particularly differentially expressed, we could procure them. The procured ontology of every target gene was categorized based on their functions, including their cellular components, as I told you, molecular functions and biological processes. The targets of MIR 1861E include the proteins which are involved in the cellular transport as well as the defense-related proteins. Okay, so this was done. Now the next part was to check the functionality of this miRNA. So far it was okay, it was only based on either on in silico tools or the analytical tools which are available in the lab. But if somebody wants to see, you show the result, how it actually works in plant. So there are two ways to work on microRNAs. Either you can overexpress or you can knock out depending on whether the microRNA you got is a positive regulator or it's a negative regulator. So either you can go for agrobacterium mediated transformation or you can also use the STTM technology to downregulate or to knock out the particular miRNA or even their targets. So we selected the sensitive variety and we introduced this miRNA. We prepared a construct in agrobacterium tumefacium, and then we overexpress this MIR in the rice embryogenic callus species. So these are the sequences. This was the construct which we use. This is PCambia 1301, which is a binary agrobacterium vector. So you can have dual selection process for the transformants as well as for the bacterial selection in the medium. And we are working with this, PCambia 1301 system for so many years, and it is well-established system in our lab. Okay, so after that, we verified and we cloned and sequenced, as I told you, and we got the results. This is the schematic of the work. Okay, so what we did, we tried to get the embryogenic callus directly from the seeds. Rather than growing them, germinating them, we got the callus from the seeds, and these callus pieces were transformed using the agrobacterium strain. And then these were put on the antibiotic selection. And you can see here the non-transformants, they are dead, whereas the putative transformant, I'm saying putative because so far we don't know. Putative transformants could grow on the medium containing the antibiotic. So after that, this callus was developed into shoot, the process which is known as indirect organogenesis. And finally, the plants were rooted and they were transferred to the greenhouse conditions. So we try to check whether the transformants are having the desired miRNA, and these results, the normal PCR and agarose gel electrophoresis, it confirmed that it consists or it contains the desired gene. So this process was completed. Then we also checked the performance of plants under the stress conditions. We introduced again both the stresses as we were talking about in initial part, and then we could see the differential expression. So all this result was also supported by different biochemical parameters, reactive oxygen species, then antioxidant enzymes. And again, we check the expression level of this particular microRNA in controlled wild type as well as the treated plant samples. And you can see here that the expression level was differential. That means the construct is working as well as the microRNA is also working. As I told you that some other parameters were also studied, including biochemical. So we studied the ion contents, particularly sodium, chloride, potassium, and the very important sodium-potassium ratio, and the results are here. So as expected, we got the differential result in terms of control or wild type as well as the transformed plants. So the precursor of top sodium responsive microRNA was successfully overexpressed in rice. particularly using embryogenic callus, and the expression level were increased where of microRNA were increased, whereas the expression level of the targets of that microRNA were decreased, and that's what we were expecting. So this study was completed in that way. And this is the crux of the story, that the sodium was responsible for inducing stresses. Then we identified the top responsive microRNA, and then the differential expression pattern of microRNAs was also checked, and this selected microRNA was successfully transformed into rice callus, and the differential behavior of transformed and untransformed plants was observed. So as I told you, we got some funding support from DST, DBT, SERB, and DABRNS for this kind of work, and that's why we could complete this. One of my PhD students, he was working on this particular work on rice. Thank you so much. These are some of the publications on microRNAs. Thank you. If you have some kind of questions, comments, or suggestions, you are most welcome. If anybody is really interested to start working or any kind of help is needed for microRNA work, you are most welcome. As I told you that we officially have a collaboration with MIT. So these kind of possibilities are of course there. Thank you one and all for patient listening. I hope it was worth listening, and if it will help you in any of the ways in your research programs. Thank you so much.
I would like to thank Dr. Binay Kumar sir for the informative session, and it was about miRNA and RNA-based researches. Very few groups are working on miRNA or RNA in this world. So because this is very costly research or something more prone to fluctuation in result. That's why most of the group are not working. But whatever you have presented, it was a very nice presentation. It was
thank you.
Everyone can understand this, and I think myself is interested to work on this topic with you.
Welcome.
And most of our colleague is also contact with you, and I think some of participants definitely contact you regarding this. So thank you very much, sir, for sharing your vast knowledge related to research and development, and teaching also. You are very much associated with teaching. Now session is open for question and answer. Any participants can drop their question in chat box. Meanwhile, I would like to ask one question.
Yeah, please.
Sir, is there any relation while we are working on rice? So is there any relation of microRNA with grain filling?
We only are working. As I told you, nobody is working on reproductive phases in rice, though many people are working in India in rice. But when I was proposing this to DST, I myself was very surprised that nobody is working on reproduction. Though the fact is that reproductive phases are more sensitive to any of the stress conditions. But probably the maintenance of stress condition is very difficult, and we also had lots of struggle to finalize the treatment stage, for how many hours the treatment should be, because if somebody is working on microRNA, the time frame is also very important. Unlike other biochemical and molecular parameters, the microRNA are the instant reaction of plants. So you must have seen that particularly for microRNA work, we only gave treatment of maybe 8 hours, 16 hours, or 24 hours. Because after that, the miRNA start getting suppressed. Because probably, nobody knows why, probably the reason might be that it is way beyond their capacity now. Some other players are getting into the picture.
Yes.
So nobody is working on grain filling, and it's a good area. You can definitely think of exploring.
Yes. Sir, there is a question from one participant.
Yeah, please.
Can mRNAs help plants to cope up with biotic stress too? Like we can use them to increase plants' immunity against invading bacteria and fungal pathogens?
Okay, very nice question, and the answer is yes. Since I told you that microRNAs are involved virtually in all the plant processes. So, and मैंने एक जुमला भी मारा था शुरू में ही कि सब कुछ जायज है प्यार और जंग में.
Yes.
So the same, goes here also. Yes, many a times, things don't go as we want them to go, and the same case happens with plants also. कभी-कभी हमें पीछे हटना पड़ता है. The same way plants also do, they attenuate their own growth just to cope up with the stress conditions. Suppose there are some plants and they are facing the drought condition. There is no irrigation or water. So immediately the roots will pass on the signals that to the leaves, because unfortunately water is coming to roots and the photosynthesis is happening in the leaves. So energy and this signaling is absolutely different. So plants have to pass on this message. So this signaling pathway is very important, and again it requires lots and lots of gene programming and reprogramming. So the same case and the same thing happens with biotic stresses also, and all kind of interaction of plants with animals as well as microbes. There are involvement of microRNA mediated responses. I hope the answer is reached.
Yes, sir. It is clear now for the participant. And again, thank you very much, sir, for being with us. I think there must be some question, but not yet reflecting in the chat box. If any query will be there, then we'll try to contact you.
No worries. Thank you once again. Thank you, the team. You have a wonderful team, Dr. Amit Datta, Dr. Kunal, Dr. Rahul, Dr. Sumira. So let's see if we can catch up sometime in person also.
Yes, sir.
I'm looking forward. And thank you once again for giving me this opportunity to interact with all of you. Thank you so much, and wish you grand success in this conference. Thank you.
Most welcome, sir. Now I am handing over this session to Dr. Shilpa Prasad ma'am for some digital content release over here, because Honorable Vice Chancellor sir is with us now, and we'll do a small celebration here. Ma'am.
Thank you, Rahul sir. As sir has just mentioned that our Honorable Vice Chancellor, Sir Dr. Raman Kumar Jha sir has already joined, and I, on behalf of Organizing Committee, heartily welcome you, sir. So please share some words of wisdom with us on this platform.
Namaskar, Jha sir.
Namaskar. Thank you very much, Dr. Prasad. I don't have anything to share because I did not hear you. So if you want to hear me and my viewpoints, those are separate than the relevant things which should be spoken on this occasion. But the common thing is, sorry that I missed it. Sorry that I was delayed, and my best wishes to those who attended, and I'm sure they have gained. I was listening to last 5 or 10 minutes of yours while you were discussing things on mRNA related to rice and its production and so on. So those I don't come from that field as well. So I would only say that the production of research must begin with you as the core center and the other people from biotechnology department at AUJ. That would be my endeavor. And research is not something which can be done in a day. It is a collaboration. It is a thought process. It is more of a commitment that you do for a particular field for longer time. If you do it, you still may not get results to the desired direction or in the way you thought about it. Therefore, it is a ball game of academicians without thinking of any results. So this is how it goes, and only maybe some enlightened few will have the hunch, yes, this may come, or let us see what comes. So that's research all about. Can we also get on to the level of bringing that to a startup, or bring that some economic aspect of it or value to it in terms of economic or commercialization of the product that you do is the next level to which we are all thinking. University would encourage everyone to take things forward. My best wishes once again, Dr. Amit Datta. Sorry once again, personally and officially, for not having attended your sessions for long. Thank you. Over to you. I'm here now. If you want something to be done, we'll do it. Thank you.
Yes, sir. Thank you so much. So now I request our Honorable Vice Chancellor, Sir, Dr. Kunal Kumar and Dr. Dhananjay Pandey to kindly please release the digital version of our souvenir and abstract proceedings for ICCOB 2021. As Registrar Sir has also joined the session, so please, sir.
Thank you, madam. I'm with you.
Yes, sir. Thank you.
Congratulations, MIT Biotechnology Institute and Department.
Congratulations, everyone. Thank you,
sir.
Thank you, sir.
Thank you, sir.
Thank you so much, respected Vice Chancellor, sir, respected Registrar, sir, Kunal, sir, and Dhananjay, sir. Now I request Rahul, sir, to please continue with the oral presentation session. Please, sir.
I would like to invite all oral presenters here. Now, session is open for your presentation. There is a 10-minute limitation for you, 7 plus 3. 7 minutes, within 7 minutes, you can complete your presentation, and for last 3 minutes for the discussion purpose and question answer purpose. And so first I would like to invite Dr. Bindu Sri Baghel, School of Sciences, Maths University, Raipur, Chhattisgarh. Bindu Sri ma'am, please share your content here. Dr. Bindu Sri Baghel. So I think due to technical issues or might be, I would like to invite a second participant, Dr. Kumari Rekha, University Department of Zoology, Tilka Manjhi, Bhagalpur University, Bihar.
Thank you, sir.
Yeah, welcome, ma'am.
Good afternoon to all of you. First of all, I would like to thank the members of the organizing committee for providing me an opportunity to discuss my research work. The title for my research work is Jambolin as remediable in stabilizing proper function of testis of alloxone-instigated pale mus musculus. Introduction. So, diabetes mellitus. Diabetes mellitus is a condition in which the pancreas no longer produces enough insulin.
Rekha ma'am, please share your screen.
Okay. I've shared, ma'am.
The presentation is not visible to us. By that time, I would like to introduce the judges for our this oral presentation session. Dr. Jaita Chattopadhyay and Dr. Kunal Kumar will chair the session of oral presentation.
Is it visible? Is it visible, ma'am?
No, ma'am, it's not visible.
मैं बोल रहा हूँ राहुल सर, ठीक है? Amit sir, you are muted.
To kindly please judge the candidate, first of all, for the oral presentation. So, Dr. Jaita ma'am and Kunal sir, are you ready for oral presentation so that we can start?
Yes, sir, we are ready. We are ready.
Kunal sir, we can start the presentation?
Yes, sir, we can start.
Okay. So, by the time Dr. Kumari Rekha, kindly please share your screen in the full, in the slideshow mode. Put it in slideshow mode.
Are my slides visible, sir? Yes, ma'am, it is visible. Excuse me, sir.
Yes, ma'am.
Are my slides visible?
Ma'am, you can find in the very top corner, that is from current slide. Yeah. In the right, just click in the right from current slide.
Are my slides visible, sir?
Yes, slides are visible. Kindly make it full screen.
Rekha ma'am, are you hearing?
Yes, sir.
Okay, now it is
Yes, sir, I
visible,
can hear
ma'am.
you.
You will get 7 minutes followed by question answer, and it will near about not more than 10 minutes. So kindly please complete your presentation before the time.
Okay. The title of the presentation is as remediable in stabilizing proper function of testes of alloxone-instigated male musculus. Now, diabetes mellitus. Diabetes mellitus is a condition in which the pancreas no longer gives enough insulin or stop responding to the insulin produced, so that glucose in the blood cannot be absorbed into the cells of the body. Symptoms include frequent urination, lethargy, excessive thirst, chosen black plum for my experimental work. The black plum or jamun or jambo or Sazerain cumini is commonly known as Malabar plum. It is an evergreen tropical tree in the plant. The experimental animal which I have chosen for my research work was Swiss albino musculus. The meta more or less Swiss albino musculus is a mouse brains which is widely used to manage research.
Dr. Rekha, kindly make it full screen.
Like,
kindly make your presentation full screen mode.
Is it visible? Oh, sir.
Not yet.
It is going full.
Okay, please continue. Please, I think there could be some technical glitches at your end. Please continue with whatever mode you are being able to. Please continue.
Dr. Kunal sir, alternatively, because we already have those PPT, so can I share at my end?
Please try to do that, Dr. Dhananjay. Dr. Rekha, we are trying to share your PPT from our end because I think there could be some glitches at your end. Yes, Dr. Dhananjay, can you please do that?
Yeah. My slide is visible, sir? This slide is visible?
Not yet, sir.
Now it's fine.
No, sir.
Now it's okay.
Not yet, sir.
At my end, it's showing shared.
Okay, then should I ask the Dr. Rekha to continue sharing at her end?
Yeah.
Yes, Joyita ma'am, are you able to see the screen of Dr. Dhananjay?
No, sir.
Okay, Dr. Rekha, please try sharing at your own end. Yes, Dr. Rekha, please continue.
The slides are the objective behind the
Rekha, Dr. Rekha, is it possible? Are you facing some network issues? So do one thing, you just switch off your camera. You just switch off your camera and after that clear the screen.
It's okay. Objective behind her work is to elevate the ambulance and edible stabilize the function of this of zone integrated muscles. the instruments used during the research work. This is the needle used for purpose. The girl used for reduction of diabetes. This is touch of the size humani. The same. This is the powder of the same. Now testes. Testes are the sperm. They likely seminal form the bulk of each test. Established in the structure called seminiferous tubules. The seminiferous tubules are Group 1, Group 2, and Group 3. Group 1 included control mice. Group 2 included mice made by monohydrate insulin sensation. Group 3 included the diabetic mice fed with black plants with the food.
Dr. Rekha, you're not able to hear you. Joyita ma'am, are you able to hear Dr. Rekha?
No, sir. I also can't hear.
Hear you. There is a issue. I think I'm seeing the issue, sir. The methodology used for the experiment work was to 9 suspects. The morphological assessment of the sperm were performed. They were total sperm count, live sperm count, mobile sperm count, group 2 that is diabetic, group 3 that is black plum seed powder fed were simultaneously compared with the control one to establish the sequential changes in the sperm profile and sperm morphology. Assessment of the sperm were performed by hemocytometer. The total sperm count, it was calculated by the formula. Sperm count is equal to dilution into count in 5 squares into 0.05 into 10 power 6. Live and dead sperm counts were performed by a drop of 3% eosine stain was mixed in epididymal sperm suspension with the help of a glass rod. Again, a drop of 10% nigrosin stain was mixed to it. Nigrosin stain produces a dark background on which the sperm stands out as a lightly colored object. Normal live sperm excludes eosine stain and appears whitish, whereas the dead sperms appears pinkish color. The sperm motility is calculated by total number of sperm into 100 by motile sperms plus non-motile sperms. The results if they are control or normal range, sperm number decreases, the sperm motility decreases, and sperm motility in the case group increase. In case of group 3, the sperm mobility decreases, sperm motility decreases. Here is a table showing the assessment of the sperm parameters of various groups. The various sperm parameters were sperm count, sperm motility, sperm, group 1, group 2, and 3. Here is the table showing the assessment of the sperm parameters of various groups. The various sperm parameters were sperm count, sperm motility, sperm, group 1, group 2, and 3. There is another table showing analytical report of morphological abnormalities. The parameters used were sperm without tail, sperm without head, sperm without hook, sperm with double head or tail of group 1, group 2, and group 3. There is a discussions and conclusion. Fertilization is directly linked to the glucose metabolism. Glucose is required specifically for sperm fusion to oocytes. For successful fertilization, as well as to ensure maintenance of viability of embryo or proper glucose metabolism. Diabetes is associated with reproductive impairment in male with widely reported alterations in testicular function and spermatogenesis. Several other severe morphological effects are also reported, that is low motility, structural deformities, less efficient in fertility males. As the finding, this powder has protective effect on different tissue, including and immediately effect on the sperm profile. Black plum seed powder treatment increases the number of spermatogenic cells in mice of group 3 when compared to that of diabetic animals, that is group 2. It was found to be increased with the exposure period. Black plum, by correlating its already identified phytochemicals with their described mechanisms of action, so that some compounds target multiple metabolic pathways, thereby becoming potential pharmacological tool. There are the differences. Thank you. Now I am open to questions.
Okay, Dr. Jyoita ma'am, would you like to have any questions from the participant, Dr. Rekha?
Thank you, Dr. Rekha. Although I couldn't hear your voice properly, I have just one question. Do you have any recent time reference? Because the reference you mentioned in your presentation, that was in 2000, I think the recent one is 2003 or 2006, like that. So do you have any reference like around 2018 or 19, similar to your work?
Right now, ma'am, right now I have references of year 2014 and 2012. That is Kamal 2014 and Ramaya 2012.
Okay. Thank you, ma'am.
Okay, Dr. Rekha, I have one question. What is the purpose of using alloxan in your study?
Alloxan is a chemical. It is an analog of glucose analog, which is used for induction of diabetes in case of experimental animal.
Okay, and why did you take this particular jambulan for your study? What are the active ingredients in the material?
Various phytochemicals which are responsible for anti-diabetic properties in jambulan. It is the beta-cytosterol, arginine, various flavonoids are also present, which are responsible for their anti-diabetic properties. That's why I've chosen.
Could you find out any, out of all this compound that you have mentioned, could you find out a specific phytochemical that is responsible in reducing down the cases of diabetes?
No, sir, I haven't found it. Through various earlier research papers I have gone through that research paper, and I came to know that these phytochemicals are responsible for the anti-diabetic.
Okay, so what was the main purpose of your study? Just to reciprocate your result to higher animals, or you are happy with your the animal that you have taken?
Sorry, sir, this question.
Like what is your next step of your work? After this, where you will end up?
Oh, to apply effective property on human also. I want to whether it is effective to human or not. I would like to continue on that.
Okay, Dr. Rekha, thank you for joining us. Thank you, Dr. Rekha. Now we'll move on to the next participant, Dr. Rajendra Kumar Singh. He's going to present on preclinical toxicity studies of neem in mice and rats. Dr. Rajendra Kumar Singh, please continue. Yes, Dr. Rajendra, please continue.
Dr. Kunal, I am sharing the video now. You just discuss with RK Singh to present. I am sharing due to that network glitches. I am sharing all video.
You are sharing the PPT of Dr. Singh?
Yeah, everyone video I will share. So
you are saying the PPT. Yeah. Okay. Dr. Rajendra Kumar Singh, you can stop sharing from your end. We are trying to share the PPT from our end. Dr. Amit Datta will do that. Dr. Rajendra Kumar Singh, can please stop sharing. Dr. Rajendra Kumar Singh,
yes.
You can continue with your presentation now.
Okay, please next. This biological cell membrane exhibit highly specific interaction with the drugs, antibiotics, viruses, etc. due to the surface glycoprotein with specific sugar moieties, immunological interaction including mast cell and other excitable tissue interactive responses depend on the glycoprotein receptors. Number of quinoline alkaloids found in plants have a glycosylase inhibitory activity. They can modify glycosylation physiology and alter the membrane interaction with the ligands. Aja directa indica, commonly known as neem. The family Meliaceae is commonly known as neem, which is found in various parts of India and neighboring countries. The leaves is used in the Ayurvedic system of medicine for treating antiseptic, anti-inflammatory, anti-ulcerogenic, bronchial asthma, antioxidant, immunomodulatory. Next. These drug administration is a directa indica. Fresh green leaves were collected from the Institute of Medical Sciences, Banaras University, Varanasi. After the approval of Institutional Animal Ethics Committee, the present study was conducted in the Department of Pharmacology, Institute of Medical Sciences, Banaras University, in bred mice and rats. Next. The fresh green leaves were separated from the stems and crushed with the help of pestle metal to make a paste and then squeezed to get a juice. The juice was directly used for experimentation because Ayurvedic literature suggests in this form. Next, please. The toxicity study is the sample study design used for the toxicity is commonly mice and rats, and OECD, as per the OECD guideline, Organization for Economic Cooperation and Development and WHO guideline, acute toxicity is 423, that is the single dose administration, and second is the subchronic toxicity 407, repeated dose 28 days, oral toxicity study in the rodents. Next. The acute toxicity albino mice, Swiss, 5 male and 5 female, weight 20 to 40 gram, and dose range is single dose, oral administration 10, 100, 200, 500, 1000 and 2000 milligram per kg plus control. Observation is 1 hour to 14 days. Next. This is the drug administration. Next. The result of the acute toxicity, all the animals treated with the different doses of Aja directa indica leaves showed a normal behavior and no mortality was recorded up to the 14 days, except 2000 milligram per kg dose, 10% mortality was recorded. After postmortem and histopathological examination was performed, actual root cause of mortality is higher exposure of dose. Next. The subchronic toxicity, albino rats with strain, 5 male and 5 female, weight is 100 to 150 gram. Dose range is single dose oral administration, 50, 100 and 200 milligram per kg plus control. Observation period is 28 days. Next. These are the subacute toxicity body weight of animal, the initial weight and first week, second week and third week and fourth week. There is no significant difference of the difference between the control and treated animals. Next. These food and water intake in the composition weekly treated Azadirachta indica, control versus Azadirachta indica 2000, 1150 milligram per kg. There is no significant difference in water intake and feed intake. Next. These are the hematological parameters and RBC, WBC, hemoglobin percent and differential counts. And there is no significant difference was observed in control versus treated. Next. Next. This is the subacute toxicity weight of the vital organs collected from the last date that treated with Azadirachta indica in 28 days. And there is no significant difference in heart, kidney, adrenal and spleen and liver weights. There is no significant difference control versus treated groups. Next. The acute toxicity study mice treated with Azadirachta indica up to the 1000 milligram kg caused no mortality and normal behavior. The animal treated with 2000 milligram per kg body weight, so 10% mortality was recorded. And subchronic toxicity, all the 3 doses showed normal behavior and no change in the feed, water and hematological and biochemical parameters in comparison to control animal. And vital organ weights, no significant difference between the control versus treatment. Renal and spleen, pure isolates of active principles need testing towards identifying drug therapy and pharmacological properties of medicinal plants. Thank you.
Thank you, Dr. Rajendra. Now I request Dr. Joyita to kindly ask the queries she has.
Yeah. Thank you, Kunal sir. Thank you, Dr. Singh, sir. Nice presentation. I have only one query, that what is the long-term significance of the present research work?
Actually, the NEEM is working in the different aspect for the anti-inflammatory and hypoperfusion and blood vessels perfusion. There is the significant effect of the anti-inflammatory activity and significant activity in other parts. This study is necessary for the toxicity study. If the maximum dose that cannot be harm safety and efficacy of the neem juice. Actually, the number of compounds that can be isolated from the neem, there are the different criteria, but the juice is no harmful for this for the animal study. That's why we are using. The Ayurvedic literature that shows the juice is used. Okay. If you go for a isolation of compound, the number of compounds they can use in a different criteria for modern science.
Okay. Thank you, sir.
Thank you very much, madam.
Yeah.
Dr. Rajendra, I have one couple of questions. Like, yes. What was the motivation behind your study? Like, as you said that it is widely recommended in Ayurveda that we can have these, neem juices.
Actually, the Ayurveda literature is the modern science is not saying like that. There are number of extract and number of compounds we can isolate it and we can identify it. In pharmacological aspect, we can identify if the chemicals is there. If any parts of the plant that have a number of activity is present. If the isolate is the different functions in the number of patents from United States from the another content. So we are valuing the Ayurvedic person saying these activities are this is the fruitful for the human society. That's why we are using that.
Okay. Another question is what are the different bioassays you have carried out in your work for toxicity studies? Because that was not getting completely reflected in your methodology. So I'd like to have a response.
If the bioassay can do for the receptor activity, we can do that. That is not done in this. That's why we cannot answer it.
Okay. Thank you, Dr. Rajendra, for
very
joining
much.
us. Thank you very much. Now, the next presentation is by Ms. K. Madhumita. She is going to present on comparative study on effect of vermicompost and chemical fertilizers on yield components of Oryza sativa. Yes, Ms. Madhumita, kindly present. I think Dr. Amit Kumar Datta will share your slides.
Can I share my slides, sir?
Now we are trying to share from our end. Amit sir.
Sir, can I share my own slides, sir?
Is your internet connectivity strong enough to share? Then we don't have any issues. Please share.
Okay, sir. Thank you so much, sir. Sir, is my slide visible?
Yes, it is visible. Please continue.
Sir. Yes, sir. Good afternoon to one and all gathered here. I'm Madhumita from Biotechnology University, and I'm here to present my research, my project research on comparative study on effect of vermicompost and chemical fertilizer on yield and yield components of Oryza sativa. Rice is an important crop of a Poaceae family and a major staple food that is widely consumed all over the world. Inorganic, organic, biofertilizers are the main source of replenishing the plant in agricultural soil. For the food production in the ancient times, we used organic fertilizer, but to compete the growth of a population, now we are in a situation to change through chemical fertilizer. This has occurred after the Second World War. Now, in the year of 1950, after the Green Revolution, government itself prescribed the chemical fertilizer to people for increasing the yield. But as a result, fertile soil is turned to sterile. The lack of its nutrition capacity and the toxical level is also get increases. Researchers said that the use of chemical fertilizers decreases the crop yield, soil acidification, decreases in the biological activity in soil. Soil changes even its physiological properties and the lack of nutrition even in the agricultural land. So we have turned to the organic farming. In my personal experience, in the 4 years of organic farming, it is very hard to take even the investment amount. So my family, again, not only my family, even the village members are also turned again to the chemical fertilizer. So in my request, I have made the studies as a comparative study on a vermicompost as a organic source and also chemical fertilizer. In 1 acre of land, I have selected one plot for vermicompost and the rest of it for chemical fertilizer. The entire study was carried out in Palu, Trichy district in the Tamil Nadu. The study occurs in the duration of September 14, 2020 to February 10 of 2021. Averagely, it occurs for 5 months. The vermicompost plot is considered as a plot A and the rest of the areas is considered as a plot B, but the same amount only of 0.17 acre is considered for the study. So the requirement is 1 acre is 250 kilogram. For the 1 set means 2.5 kilogram. For the 17 set, I have used 42.5 kilogram. For the first time we have used the vermicompost means the amount must be the twice. So I have twice the amount, that is 85 kilogram I have used for 0.17 acres. So before plowing, I have prepared my land with the Poaceae plant where you have seen the plant below, and the Thespesia purpurea. This is a kind of a plant that used to increase the yield, which is full of organic nutrients. It's a small, very small plant that you have seen above the side. So it has been done in some September 14. After that, the land was prepared by ploughing. The seeds. The land was prepared for spraying the seeds. The seedling was raised by the wombat method. In the September 21, the seed has been inoculated on land. The variety we have used is COR50. The duration of the crop is 130 to 135 days. The COR50 is released by the Central Variety of Release Committee of South India in 2010. It's a hybrid crop. The parentage was CO43 and ADT 38. The season was late Samba and Taladi. The grain type is medium and slender. The rice color was white fine rice. The special features is the moderate resistant to blast, sheet blight, brown spot, bacterial leaf blight, the rice tungora disease. Is the best replacement for IR20, ADT38, Co43, and it is suitable for consumption of meals, making sweets and savories. The test that was happened was actually it was released in the 2000 itself, but the committee that was released these days after several testing for 10 years, and then only they have released it. That was these were the trials that have been made. So that was I was mentioned. And the application, I directly come to the applications. And in October 17, it has been inoculated with a spacing of 15 centimeter between each plant by hand. And the fertilizer has been applied according to the table. The two plot has been divided as I have said before. The plot A is considered as a vermicompost and the plot B, and you can consider as a chemical fertilizer. Total areas covered is 0.17 acre. God. I'm sorry, some technical issues happened. Okay, this is what fertilizer for its commonly for 1 acre. I have made this chart. The total investment for this, if you use only the vermicompost, means that is 2,850 rupees only. For the first time means 5,350. That was friendly for farmers. That is what I have mentioned in it. And if you use the chemical fertilizer for 1 acre means we have to use NPK complex fertilizer, zinc sulfate, urea and potash for twice. So the total investment amount is rupees 6,015. It was a common method practiced by the delta farmers of Kaveri. Vermicompost has a large particulate area that provides a site for microbial activity and retention of nutrition. And the chemical fertilizer can accelerate the growth of the plant fastly than the vermicompost. So that is what I have observed. In the 4 years of organic farming, we have used these products only, which is provided by Tamil Nadu government itself. That is Azospirillum lipoferum, Bacillum megaterium, Pseudomonas fluorescens, Mycorrhiza acetobacter. Since the yield was low, we have observed mainly in the year of 2018 when who are the farmers using the chemical fertilizers, land was affected by the pest attack, but still the farmers who have used these organic fertilizers has been escaped from the pest. I have searched why this happens only for the organic farming. Then I conclude that from the research papers and all, that the bacteria produces salicylic acid on the surface, and so it resists the crops from the insect attack. And this is what the result I have. Excuse me. Can I continue?
Yes, ma'am.
Okay, ma'am. Thank you, ma'am. And this is what the result I have got. The weed attack is comparatively less compared to plot B. Even the pest attack too less compared to plot B. But when you commercially come again, the length of the root or the number of seeds or total grain of
Dr. Rajendra, please mute yourself.
Ma'am, can I continue? Yeah, ma'am, thank you. But when you see on a commercial angle, like the length of the stem, length of the root, or a number of seeds or total gain, the plot B that gives the maximum yield. So this is for the nutritional content. That means after harvesting, I have taken the soil from the four sites, and I have mixed it, and I have given to the Tamil Nadu agricultural portal for the testing. So the nutrient capacity that was gained in the soil is much in plot A rather than the plot B. So plot B shows a good results and in yield than a plot A. So the acceleration growth of plot A vermicompost is nearly low compared to plot B. So I have concluded that neither an organic fertilizer or inorganic fertilizer alone can result a sustainable reproductivity, because even in my study, what other research paper have said is vermicompost is only good for the yield, but when it's come to the application on a farmland, it's totally different. So the chemical fertilizer, it can be used for a better growth, vermicompost for improving the biological activity and reclining in the nutrition in the soil, and the bacteria fertilizers, disease-resistant crop can be produced. So by considering all the points, we have made a chart for spraying the fertilizer. That means a chemical fertilizer is also sprayed, and also a vermicompost and a biological fertilizer. That means a bacterial fertilizer also can be sprayed in a balanced amount, not on above the level. So now in the year of 2021 on September, I have applied this. So still the project is ongoing. So with a blessing, I hope that I will get a good result. So what the challenges of 2021 faced by the farmer is over rainfall. So the crop has been fall on the ground that I have mentioned on this two pictures. So the farmers again face another problem. So still we have to help the farmers for giving the better yield. So we have to think even on a commercial angle and also fertile to the soil. That's beneficial for both the one. That was a good research. I believe in it. So thank you for the wonderful opportunity provided by Amity University. I hope that I have satisfied all the audience expectation apart from my fulfillment. Thank you.
Yes, now I request Dr. Joyita to raise questions.
No, sir, I don't have any question to ask.
Yes, ma'am. Please, ma'am.
Okay, okay, and Ms. Madhumita, I would like to know. I'd like to know what is the novelty of your work.
I have to suggest to my farmer because I was in the rural area, very rural area, who is depending only on the agriculture. So what have been seen is after the year of 2000, they are using fully chemical fertilizer. When we are studying, I was in the UG and all, they will say the chemical fertilizer can turn our soils fertile to sterile. So I want to make an application on I was the one who suggests the farmers to use the. Oh my God.
Okay, Madhumita, I have a one suggestion to you.
Yes, sir. Please,
Try incorporating that in your work. Like
yes,
you have used a particular plant for your study, right?
Yes,
Now you can do a molecular level study in identifying the plant exudates released. Okay,
Okay,
every
sir.
plants are known to release some chemical compounds through their roots. Okay,
Yes,
and
sir.
try to find out the kind of chemical reaction going on between these plant exudates and the compound present in your vermicompost. See what kind
Yes.
of chemical reaction is there. Further, you need to
Okay, sir.
use FTI analysis and other bioassays and look into possible compounds being formed during this chemical reaction and how it is affecting your crop yield. Okay, and
Okay, sir.
probably you have not incorporated the result where you have used the chemical fertilizers along with the vermicompost. That result is missing in your presentation, right? So kindly try to work on that aspect also. Okay, so thank you, Madhumita, for your presentation and joining us today. Thank you. Now our next presentation is by Dr. Bindu Sri Bagan. She has joined the panel. So I request Dr. Bindu Sri Bagan to continue to start her presentations on protective effect of L-DOPA against induced testosterone reproductive organs in female Japanese squirrel. Dr. Bindu Sri Bagan, ma'am.
Yes, sir. Yes, sir.
Yeah. Please continue.
Yes, sir.
If you face any problem in sharing or PPT, we'll do that.
Sir, I can share.
Okay, ma'am.
I'm sorry, sir. I can't share my slide.
Dr. Amit Kumar Datta, sir, can you please do it? Yeah, Dr. Datta is sharing the screen on behalf of Dr. Baghe. Yeah. Yes, madam, please continue with your presentation.
Yes, sir.
Yeah.
Good afternoon to all respected delegates and all the participants. I am going to present my work on the topic is protective effect of L-DOPA against sapermethrin induced toxicity on reproductive organ in female Japanese quail. Sir, please next. These are the contents: abstract, introduction, materials and method, experimental design, results, discussion, and conclusion. This slide shows abstract of my work. Sapermethrin is an insecticide which is used to pest control all over the world. It is also considered as one of the endocrine disruptor chemical. It is considered as potent neurotoxic compound associated with the dopaminergic neurodegenerative activity. The present study was designed to test whether administration of L-DOPA can protect toxic effect of induced by sapermethrin on reproductive condition in Japanese quail. The scientific name is Cottonix japonica. The adult female Japanese quail were divided into 4 groups, 6 birds in each group. Group 1 is received saline water, and group 2 received sapermethrin.
1 minute, madam. Madam, can you put your camera on? If it's possible for me, and put your video on, camera on.
Yes, sir.
Yeah. I request other participants also during the presentation, try to put your camera on.
Okay, sir.
You need to slightly align your camera, ma'am. Slightly align your camera.
Yes.
Yeah, ma'am. Yeah. Thank you very much. Thank you. Thank you.
Okay, sir.
Please continue.
Yes, sir. All the treatment were given for the 30 days. Results showed that treatment with sapermethrin caused significant decrease in follicle weight and number of the follicles, while L-DOPA treated group showed significantly increased all the parameters when treated with sapermethrin plus L-DOPA. Exhibit nearly normal appearance of the follicle weight and the size of the follicles compared to other treated group. We observed decrease the number of follicle in comparison to control. It may be concluded that L-DOPA might be a potent protective agent against the sapermethrin-induced toxic effect on the reproduction in Japanese quail. This slide shows the introduction about my work. We all are aware know about the substance, natural or synthetic, which can alter the function of endocrine system, and this type of chemical is called endocrine disruptor chemical. This alteration may be affect the normal health of organism and adversely affect the whole population. I have selected sapermethrin pesticide. It belongs to the pyrethroid group. It's widely used pesticide in agriculture and domestic pest control area. It's also considered the endocrine disruptor chemical. Sapermethrin is considered one of the potent neurotoxic compound that cross the blood-brain barrier and affect the nervous system, and also most relevant pesticide involved in the Parkinson disease. I have select second chemical is L-DOPA. L-DOPA is a precursor of dopamine. It increases the dopamine level in the body and also treating the disease of Parkinson disease. Dopamine is a product of L-DOPA metabolism and was also found in the process 5 free radical scavenging activity and strong antioxidant capacity. All the above facts indicate the possibility of L-DOPA is recovering or minimizing the physiological metabolic alteration by the cypermethrin administration in Japanese quail. It is the basic of experiment in my work. This slide shows use of cypermethrin in different area. Cypermethrin used in agriculture field in pest control and also household pest control, vegetable and flowers, cracks and cervix, mosquito net, and domestic parasites control. Now come to the material and methods. I have selected experimental animal is Japanese quail. These are the features of this animal, why I select in my work. It's locally known as bater. It's indicate to native to the Japan and nowadays found in different other countries like India, China, and Korea. It is widely used for the egg production, meat production, and the laboratory animal. Its maturation time period is 6 to 7 weeks. And the sex difference appeared about the 3 weeks of the age. The weight of the male bird is 100 to 60 gram and female bird is 150 to 190 gram. Male bird are characterized by the rusty brown throat, breast feather, and the presence of cloacal gland, and female have a pale breast feather that is speckled with a dark colored spot. Methods. Female reproductive organ, I have selected follicle weights of the quail were taken by the using of electronic balance and the size measured with the help of vernier calipers. This slide shows the experimental design. I have taken female Japanese quail and divided into 4 groups and selected these are the parameters: follicle weight, number of follicles, and size of the follicles. This slide shows the photographs of follicle. When I treated with cypermethrin, the follicles are the decrease and also number of the follicles decrease. When treated with L-DOPA, the photograph is L-DOPA treated group is showed significantly increase the number of follicles and also the increased size of the follicles. When treated with both chemical, when treated with both chemical, the follicles are near about the control group. These are the results of my work. This graph shows follicles weight and the number of follicles. When treated with cypermethrin, it significantly decreased the value of when compared to other treated group. Same result was found in follicle size. It was found interestingly result when treated with both chemical, the value was near about the control group. Sir, please next slide. This slide shows the result. Follicle weight and size and number of the follicles cypermethrin treated group showed significantly decreased value when compared to the control and L-DOPA group. L-DOPA treated group showed significantly increased value of follicle weight and size, whereas the number of follicles were no significant different when compared to control. Cypermethrin plus L-DOPA treated group showed significantly increased value of follicle weight and decreased number of follicle in comparison to control group. This slide shows discussion. On the basis of the observation of the present study, it has been apparent that administration of cypermethrin, L-DOPA and their synergism produce variable effect on the reproductive condition, male and female reproductive organ, serum testosterone, testicular acid phosphatase, sexual maturation, and on egg parameters, egg laying, production, quality, hatchability, motility, fertility, and embryonic development in Japanese squirrel. Sapermethrin induce impairment in reproductive indices and cause reproductive disruption. On the other hand, L-DOPA induce and maintain reproductive condition. L-DOPA is combination with sapermethrin reduce the disruptive effect of sapermethrin on reproductive indices in Japanese squirrel. Yes. This slide shows the possibility of this outcome could be explained with this diagram. The diagrammatic representation of possible mechanisms showing the protective role of L-DOPA against the sapermethrin-induced toxicity. Sapermethrin is induced dopaminergic neuron in brain and also affect the tyrosine hydroxylase activity and aromatic amino acid carboxylase, and then decrease the dopamine. And dopamine is affect the anterior pituitary releasing hormones, gonadotropin releasing hormones, and affect the gonads. But L-DOPA, when treated with L-DOPA, increase the activity of TH activity and aromatic amino acid decarboxylase.
Madam, you have crossed your time. Please, finish in
Yes,
another
sir.
1 minute.
Yes, sir. And increase the dopamine level and affect the gonads. On the basis of above experimental finding, it may be concluded that L-DOPA might be potent protective agent against the sapermethrin-induced neurotoxic toxic effect on the reproduction and develop in Japanese squirrel. The possible mechanism may be explained on the basis of ultrasound change occur on neuroendocrine gonadal axis. However, it needs further study on molecular basis. Thank you, sir.
Thank you, madam. Joyita ma'am, your responses, please, if you have any queries.
Yeah. Thank you, ma'am. Thank you for your nice presentation, and it was quite informative session also. I have one question that can you tell me the chemical composition of L-DOPA?
Yes, ma'am.
What is the chemical composition of L-DOPA?
L-DOPA is a precursor of dopamine.
Okay. And what was the concentration of L-DOPA which was administered?
5 mg per 100 gram body weight.
5 mg per 100 gram of body weight.
Body weight of bird.
Okay. 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 Thank you. Thank you.
Madam, Bindushri ma'am, I would like to know, why did you choose this particular, animal model for your study? Is there any specific reason?
Yes, sir. It's easily maintained in laboratory, sir, and also size. सर बर्ड का साइज भी इतना पोर्टेबल है कि उसको लैब में यूज कर सकते हैं सर, इसलिए ये एनिमल सेलेक्ट किया मैंने.
Okay. Now you're telling in your result that it is L-DOPA which was having some, positive impact in increasing the follicle size. Now you should also find out that is not reflected in your work. Is it L-DOPA or no, the L-dopamine which is actually working? Because you should also find out the L-dopamine concentration, if any, in the tissues and the body fluid that you need to work out. Okay. So instead of that, instead of administering L-DOPA, you can use either L-tyrosine or L-dopamine to see the synergistic effect. Okay, this you can use that also. You can apply that as well. Okay, thank you, ma'am, for the presentation. Thank you for joining us. Now the next presentation is by Mrs. Samvedna Singh. She's going to present on identification of potential prognostic markers in pancreatic cancer using an integrated approach of gene expression profiling and network analysis. Mrs. Samvedna Singh, I would request you to present your PPT.
Thank you, sir. So shall I share the screen?
Yeah, please share. Please share.
Please let me know, sir, if it is visible.
Yeah, it is visible.
Thank you so much, sir.
Good evening, everyone. My topic of presentation is identification of potential prognostic markers in pancreatic cancer, integrated approach of gene expression, profiling, and network analysis. Pancreatic cancer is one of the leading cause of deaths and morbidity due to cancer. The 5-year survival rate of cancer, pancreatic cancer, is less than 10%, and there are no prominent biomarkers already figured out, and that is why the patients who get diagnosed are diagnosed with the very late state of the pancreatic cancer. That is the end state, or the stage 4th of the pancreatic cancer. The present study aims to gain a better understanding of the mechanism involved in the onset of progression of pancreatic cancer, as well as to suggest some prognostic markers for the detection and prognosis of the disease. Gene expression changes the development and progression of the diseases, and that is why it is a very useful method. Gene profiling is a very useful method in order to identify the disease pattern and the involvement of entities in the disease. We identified 464 differentially expressed genes and the predictive usefulness of significant genes, as well as the pathways they are enriched in. The methodology we followed is first data collection, second is expression analysis, differential methylation analysis, ontology and pathway enrichment analysis, and then integrated protein network analysis, module analysis, and selection of hub genes. For the first 4 set of analysis, we have used the programming language R, and for the next, we have used the softwares like String and Cytoscape. This is the table of the result. We selected these 3 data sets from Geo, and we can see the total number of genes are very high. However, we have figured out a few differentially expressed genes. When I say differentially expressed, I mean the ones which are either showing the upregulation, that is overexpression in the particular disease, and when I say downregulated, they mean they're showing the suppression of the genes and showing the expression level less than their usual expression in the disease, in the normal patient also. These are some volcano plots we have used for the visualization. We can see here right and left and right direction. The left direction shows the negative fold change value, which means the expression of the genes are higher, and the right hand side shows the lower expression of the genes. Amongst the comparison, the right one shows more genes because it was having more samples. This is the figure of the upregulated genes, when I'm saying the left one. This is the figure of the left number of genes, and we can see that there are 405 upregulated genes, and we can see that the hubs are forming in the protein network. So from the gene study, we are moving towards the protein study so as to identify their interaction amongst each other. So here we can see that the upregulated genes are forming a better network than the downregulated genes, which means the overexpression of the genes are highly prominent in the diseases like pancreatic cancer. This is the complete network. It shows 394 differentially expressed proteins, and the confidence score selected is 0.4. We can see some hubs, but because we also want to identify the topology of the network, we want to understand how strong the network is. We are studying it further with the app called Cytoscape. This is the table of the main genes came into the hub. This is fibronectin genes. These are some values given by the string. Then after that, as I already mentioned, that the bigger network we are studying in the topological order, and that's why we are using Cytoscape app called MCode. So this is the first module, which shows and gives us the score of 21.692, and the number of nodes is 282. So it means that it shows a very high stringency and very high closeness and centrality amongst themselves. So this indicates that we can study this network in diseases like pancreatic cancer. We also have taken into the consideration the ontology of the genes so as to identify which genes are performing the roles in which another diseases or say pathways, and what are they doing in the pathways? Like, for example, are they regulating it? Are they suppressing the normal pathways? So we have found out that there are genes who are involved in extracellular matrix organization. They are involved in age-range pathways. They are involved in PI3K-AKT pathways, and they are involved in RAS oncogene pathway or pathways of cancers. This is the second module. It gave the score of 6.53. And it also contained few genes and proteins which were included in the main pathways like cytokine signaling of the immune system and the pathways which affect the immune system. Also type 1 diabetes mellitus pathways, which indicates that these genes can also play a role in such diseases like type 1 diabetes mellitus, etc. This is the third module, which shows that the genes involved in these modules, they are involved in focal adhesion, tight junction, and GT-phase activity, Rho GT-phase activity, etc. Discussion. Pancreatic cancer is a complicated disease. Why I say complicated disease? Because it does not happen, or it does not revolves around just one gene. It revolves around networks of genes, and if we study the network of genes, we also study the pathways the genes are involved in. And then if we study these rows of genes which are present or absent in the disease, we can identify the regulatory network which is forming during the patient is having that disease, and thus we can work on those markers. The biological processes, as I talk about, it included cell adhesion, it included ECM receptor interaction, and various pathways of cancer like proteoglycans in cancer, etc. So the promising molecular markers I'm suggesting here are CABP4, GAT5, ACE2, MMP1, fibronectin-1, EGR2, and studying these and can be useful for effective therapies for in future for pancreatic cancer. Thank you, sir. Thank you.
Thank you, madam. Now I request Dr. Joyita to ask queries.
No, sir, I don't have any question to ask.
Okay. Samvedna, madam, like you have found out many molecular markers through your study, which I believe are surface expressed proteins found on the pancreatic cancer cells, right?
Right, sir.
So now I would like to know, did you find some common molecular markers which are also present in the cancerous cells of other organs through your literature study?
Yes, sir.
Is there any similarity?
Yes, sir. Of course, there are various genes which are involved. Also, I found out that few of genes were already discovered in pancreatic cancer, but not as much. That is why I opted for microarray studies. Also, it has been found that it was involved in diseases like breast cancer, prostate cancers, etc. Yes.
Okay. So we can go ahead with monoclonal bodies mediated, diagnostics of the diseases and therapy, right?
Right, sir. For the essays, right.
You have found out the the specific molecular markers present on the
right.
cancer. Okay, a good work done, Mrs. Samvedna Singh. Thank you for joining us in this conference.
Thank you for the opportunity, everyone.
Yes,
Thank you.
Now the next presentation is by Mr. M. Srinivas. She's going to present on homology modeling of flap endonuclease 1 protein of Leishmania and its molecular docking with novel inhibitors. Yes, Mr. Srinivas, please continue with your presentation.
Yes, sir. Thank you, sir. I can share my slides, sir.
Yeah, please continue.
Is it visible, sir?
Yes, it is visible.
Okay. Good evening, everyone. I am M. Srinivas, and the topic for my today's presentation is homology modeling of flap endonuclease 1 protein of Leishmania donovani and its molecular docking with the potential novel inhibitors. So starting with the presentation first, what is leishmaniasis? Leishmaniasis is a parasitic disease and is caused by an intracellular protozoan from the genus Leishmania. Probably that is Leishmania donovani and Leishmania infantum or Leishmania chagasi that are commonly the species that are indicated in the disease. Now, leishmaniasis is transported by a fly, that is a sand fly, and it is a tropical and a subtropical disease. And it is carried by the bite of a sandfly of mainly species that is Phlebotomus and Lutzomia. And they are from Europe, and they are found in Europe, Northern Africa, Middle East, Asia, and parts of South America. Now, leishmaniasis can be of three clinical forms, mainly cutaneous, mucocutaneous, and visceral, that is involving skin, mucous membranes, and visceral organs. Now, among these three, visceral leishmaniasis is the main cause is the main type of leishmaniasis generally seen, and it is the only cause next to malaria for the majority of deaths worldwide. And according to WHO, more than 350 million people worldwide are at a risk of leishmaniasis, and in that currently 12 million active cases are from more than 98 countries. Now, the life cycle stage of a leishmania, it starts in sandfly. Sandfly takes it up from a blood meal. That is, if sandfly bites an infected person, then leishmaniasis travels into the gut, mid-gut of the mosquito, and then it differentiates into amastigotes. They transform into a promastigote, and they remain dormant in the proboscis. Now, when this infected sandfly, if it bites a human, that is a healthy human, then these promastigotes that are lying dormant in the midgut, they travel into the bloodstream. These promastigotes transform into amastigotes, that is the active forms inside the macrophages of the person. And these amastigotes then rupture the macrophages, release themselves in the bloodstream, and affect the neighboring macrophages or other cells. And in this case, this is how it is spread throughout the body, and it forms black spots of the skin and also affects the visceral organs. So we are targeting, our main target protein here is flap endonuclease 1. Flap endonuclease 1 is an enzyme, and it plays a critical role in maintaining the genome stability and integrity of the Leishmania or the Leishmania protozoa. And it is a structure-specific nuclease with 5-flap endonuclease property and 5-3-exonuclease activities. It basically, the function is that it leaves the 5-overhanging flap structure that is generated by the displacement synthesis during the DNA replication part. And flap endonuclease also has RNase H activity, and it is actively involved in replication and repair of a recombinant DNA and in repairing of a mitochondrial DNA. Further, now we come to the objective of our part, the objective of our topic. In that present study, what we have done is we have refined and modeled the flap endonuclease 1 protein on our own. For this, we use two different templates. That is for homology modeling, two different templates were used. PDB ID, one was 1UL1 and the second was PDB 5ZOD. Now in this, after that, we did the molecular docking of this molecule with the novel inhibitors. And then we screened some potential drug compounds through the online databases. And for this purpose, the docking and the binding interactions of proteins and ligands were done through Accelerate's Discovery Studio 2.1 and Swiss PDB software. And after all the docking processes, the dock and the leak scores were analyzed to find the best drug compound and the best dock conformation. Material method, first we had to retrieve the sequence of our flap endonuclease 1. It was obtained from the UniProt protein sequence database with it had an ID of E9BIWO, and it was seen to contain a 395 amino acids in the sequence. Then we went on for template searching using our flap endonuclease 1 sequence as the primary search query. We went to the BLASTP software, used the BLASTP to screen out similar templates from the PDB database, and we got two of them was seen to have the most overlap with the flap endonuclease 1. One was the chain X of flap endonuclease 1 that is also of Homo sapiens, that is human origin, that is PDB ID 1UL1, and the A chain of flap endonuclease 1 again of Homo sapiens, and that was of PDB ID 5ZOD. And they were seen to be 53.43% and 53.61% sequence similarity they had, and had an overlap, the sequence overlap of 88% and 84% respectively. Next, we went on for the homology modeling. The homology modeling was done with the help of SPDBP, that is Swiss Modeler software. The secondary structure of a model protein was predicted so that we could know that what kind of structure we could expect after the modeling. And after the model is generated, it is evaluated and verified by servers, online servers, that ProCheck, ProSA, and ERAT. And rampage analysis was also done to check the allowed and disallowed regions that could be present in the Ramachandran plot. This is the evaluation of residues in the rampage. Here you can see that we are getting favored region as 98.4%. Residues in allowed region, that is 1.6%. 4 of the amino acid residues are present there. And in the outlier region, that is the disallowed regions, 0. So we have no amino acid in the outlier region. So it is a good model, or a good model that we have modeled, means a good structure that we have modeled. Now protein preparation, now we went on for the docking of the flap endonuclease 1 with the ligand molecules. So after modeling of the protein, it was saved and imported to X-Rays Discovery Studio. And further, we used the clean protein protocol to clean the protein and add extra hydrogens or oxygens that are required means generally. And after that, the docking process was carried out. This is the protein clean report. That was what was done to clean the protein. Complete residues, there were no incomplete residues found. Hydrogen modification was done. About 2082 hydrogen atoms were added to balance the protein. And then atom ordering was done, and we ordered atoms in 6 residues, that is glycine 2, serine 38, histidine 63, GLN 91, asparagine 138, and serine 331. Further, then energy minimization, it is done to actually stabilize our protein molecule. It was done by using the charm force field, and it has a charm force field, and the output potential energy was minus 18765.41025 kilocalorie per mole. Van der Waals energy of minus 1698.922225 kilocalorie per mole, and an RMSD value, root mean square gradient of 0.24696 kilocalorie per mole. And it was done by using the smart minimizer algorithm. Here 2 different algorithms were used. One is the conjugate gradient, and second is the steepest descent, and it was done for 1000 steps. Means it was carried out for 1000 consecutive steps. Then we did the ligand screening. We had to find some selected docking for from probable ligand molecules from the zinc database. Then for that we used we did it, and we found some ligands that are amphotericin B. It's a synthetic ligand, 2,1-propylquinoline, and chymamine B, lycocalcone A, luteolin, plumbagine, and quercetin. Apart from amphotericin B, the rest of all are natural compounds. These structures were obtained from PubChem database, and for those structures that were not available online, we draw them using ChemSketch. These are the list of all the ligands that we got, and these are the source of extractions from where they can be found. Next, these are the structures of all the ligands that we screened, and on 2,1-propylquinoline was drawn in ChemSketch as it was not available online. The structure was not available online. Then compound preparation, here with that same thing, that energy minimization was done again. The charm force field was used, and smart minimizer program was used, and that same conjugate gradient and steepest descent algorithms were used. Docking study. For docking, we had to first identify an active site. For identifying the active site pocket, we use the Discovery Studio only, and we use the ligand receptor option. Then by using the define sphere option, we could obtain the active site, probable active site, and this is the active site that was to be found where our ligand could bind. Then we went on with the molecular docking. Molecular docking was done by the LibDoc tool of the Accelerated Discovery Studio 2.1. After that, we obtained a LibDoc score and other van der Waals forces, hydrogen bonds, and other data, such sorts of data was obtained, which I will show further in the result and discussion part. And after that, after docking, we went on with the ADMET, that is absorption, distribution, metabolism, extraction, excretion, and toxicity studies. And the various properties included in ADMET like absorption, blood-brain barrier, aqueous solubility, hepatotoxicity, cytochrome P450, and P450 probability enzyme inhibition study. Now we come to the results and discussions. This is the model protein that we got over here of the flap endonuclease 1. And we also did the secondary structure of prediction. So here was the secondary structure predictions. It showed that alpha helix.
Mr. Srinivas, kindly wind your presentation another 2 minutes. Okay, you are almost paused.
Okay, sir. Yes, sir. Then this is the RC plot. RC plot shows that most of the residues are in the allowed regions. Only one was in the disallowed region. And so it was only 0.4%. So it was a good structure that we got. These are the PROSA server outputs and the energy profile plot. It was a good energy profile plot, and it is a RAT analysis to analyze the errors. The errors were only found in some of the cases, some residues, and the rest of it was fine. And the overall quality score was 87.22%. Then the molecular docking was done, and luteolin was found to be the best conformation, having forming two hydrogen bonds. And it has a binding energy of minus 85.831 kilocalorie per mole. The two hydrogen forms were between hydrogen and alanine 35, and the other between hydrogen and threonine 19. And the other such other interacting amino acids were alanine 35, serine 38, proline 81, tyrosine 33, and threonine 19. Here you can see the luteolin. The binding energy of luteolin is minus 85.831 to 1. Other molecules have good binding energies, but they are not forming much hydrogen bonds. So we took luteolin as it had the maximum of hydrogen bonding, that is 2. These are the ADMET toxicity studies. Whereas luteolin, we have a good ADMET study. It has a log P of 1.73, hepatotoxicity 1, solubility level was good, absorption 0, and blood-brain barrier level was 4. It is the graphical study of all graphical representation of all the ADMET studies of all the compounds. And here you can see that in the A log P versus ADMET PCL. And the future prospect that it can be used further in further studies, and it can be made in good use in further future. And I would like to acknowledge our Srinivas Inaganti sir of Avril Biotechnology, as he has helped us a lot in this project, and we greatly acknowledge his guidance. These are the references. Thank you.
Thank you, Srinivas. Srinivas, I have only one question for you. Like, you have found out few ligands which has the possibility to act as an inhibitor for your studies, right? So out of all these ligands which are closely related, did you find out any common function group present in these ligands?
Functional group, yes. Basically, it was like amphotericin B and liposomal amphotericin B. They all had some amino acids were common in the active site. So we saw that it has a common active sites, mainly all of the amino acids. So it was like hydroxyl groups were a lot. So that is why hydrogen bonding was found to be good in these ligands.
Okay. Now, now I ask Dr. Joyita ma'am to ask queries.
Thank you, sir. Srinivas, just tell me one thing. I found many similar works. In fact, almost ditto work in internet, like literature study. So just tell me that how your present work is different from other recent works in this